Hello, everyone. Good morning. So today is day 2 of the Symposium on Eastern Ghats. Are we live? Yeah? All right. So we'll get started. Our first speaker of the day is Dr. Anupama Krishnamurti from the French Institute, Pondicherry. Dr. Anupama has completed her PhD from Pondicherry University in '97 and has been working at the French Institute of Pondicherry for over 18 years. She's one of the few people in India who studies palynology and uses pollen and cores to examine composition and continuity of vegetative communities. These kinds of methods allow us to look into the past and understand historic change and the impact of change on communities in a way that few other methods do. So Dr. Anupama, thank you so much for accepting our invitation today. We're really happy to have you as a speaker, and we can now get you to start.
Can I share my screen?
Yes, please.
Actually, there's a lot of background noise in my street. Do you want me to wait 10 minutes? You want to go ahead?
We can hear you quite clearly.
Is it okay?
Can you
hear
we can
me?
hear the background noise.
Okay.
All right.
Okay. So am I audible?
Yes, you're audible.
And you can see my presentation?
Yes, we can see the presentation.
Okay. Okay. So first of all, I thank the organizers of this conference and especially Dr. Nandini Rajamani for taking the trouble of inviting me to this. It has been a very interesting meet, and I'm very sad that I could not actually come in person to attend it, but I attended whatever I could online yesterday, and it was really very nice to listen to all the keynote speakers and all the others, including Dr. Mathu. So it's a great pleasure for me to talk to you in this symposium. And as it says here, I'm going to be talking about how we use pollen to reconstruct the past vegetation. And I'm going to present it with a few case studies from Southeast India. And one of them is actually from Andhra Pradesh, and the other one is from Tamil Nadu, but very close to Pondicherry. So there are two co-authors in this presentation, which is my colleague Prasad and also Dr. Navya Regu, my former PhD student, who is presently a postdoctoral fellow at Manipal and an associated researcher at IFT. So we'll start with definitions. So what is paleoecology? So paleoecology is very simply the ecology of the past, and it deals with the reconstruction and study of past ecosystems, including relationships between organisms and their environment. But we don't have the organisms and the environment, do we? So we need to use proxies. And to look at the environment is always a continuum. So this we'll have to keep in mind when we go about this definition. So you could also say that it's a branch of ecology studying past ecological systems, their temporal dynamics, using fossils and other proxies. But it's also the past of the ecological systems, in a sense. So these definitions you can know a lot more from Valentin Rull and Burks, who have done quite an exhaustive papers on definitions of this field. So what are the proxies in paleoecology? What is a proxy? Any indirect indicator of what is the past environment. This indirect indicator, as I had also hinted in the previous slide where we defined, it's biotic, abiotic, and all the interactions which are therein. And what are these proxies actually? So it could be pollen, which is what I'm going to be focusing on in this presentation. But equally on the pollen slide, we find charcoal. But we won't find phytoliths because it's kind of a complementary proxy, and I won't speak much about it, but it's also a very important and powerful proxy, especially when you're dealing with, say, archaeological sites, or if you want to understand what has been the past of domesticating crops and issues like that. So a whole number of things, from tree rings to stable carbon isotopes to bones, teeth, and then other micro proxies, which are like diatoms, chironomids, and of course, plant macrofossils. But many of these are not as well preserved, and they are not preserved in as many quantities as pollen actually is. So we need to use what we call as a multiple proxy approach, which means that you can do a reconstruction with pollen, but it would also be good to, for example, look at what has been the organic matter variation with that, and look at the stable carbon isotopes. So how does the stable carbon isotope variation match with the pollen? Because that would give us a better understanding. So it's a little bit like a hologram. So you use one proxy and you do get a reconstruction, quite a robust reconstruction. But if you want something which is better resolved, more finely resolved, then the more proxies, your reconstruction is going to be so much more robust. So what we are trying to see is the variability, the response to environment change, and other things, which including resilience, because the past data will tell us what was the kind of vegetation spectrum in the past and what is it at present. And then we can look at taxa which have been present through a long time, and giving this very long baseline of what could be very useful for when you go around to actually defining sustainability. So palynology here deals with mainly the identification and analysis of pollen grains. But the stress here is on the morphology of the pollen. It is based on the wall features. So in palynology, especially, we in fact remove the cell contents, which means that we don't deal with the DNA. So usually pollen studies, people will think about DNA, but that comes, especially in the sediment context, in a different way. So we could look at ancient DNA, because the process that we use to prepare reference slides, removing the cell contents, is what happens in nature also when pollen grains get deposited in sediments. So their inner cell contents get exuded, and it's only the wall of the pollen which is very resistant to decay, and which also has all the morphological features which will allow us to identify the pollen grains to a taxonomic group, which is not always species, but we'll get to that later. So why do we actually study pollen grains? Yes, because you can identify source plants with the pollen, and in the sediments, or sometimes if you're looking at honey, or if you're looking at loads which are carried by a bee or any other insect, the pollen groups can characterize what are the plants. So when the plant is no longer there, as in the past, or when you don't know where the pollinator has gone to collect the pollen it needs, you can use the pollen extracted from there, understand it, and then get back to the plants. As I was already telling, it's produced and you can also retrieve it in large number of quantities. And one of the reasons why this is such a good proxy is that when most other vegetal fragments have decayed, pollen survive for a long time. So these are really good tools for identifying plants. And as you all know very well, and I don't have to tell you this, pollen is meant for dispersion. So biologically, it's something which is meant for dispersion. So it is produced in large quantities, it is well dispersed, and also because of its wall properties, it's quite resilient to lab treatments. So this is why pollen grains are very important in various studies, including in reconstructing the past vegetation history. But there's always caveats when you use any method. As I said already, you can't identify plants at the species level. And pollen preservation, of course, generally it's very good when it is not, when the conditions are not arid, when you have non-oxidizing conditions, when you have material like clay, so you can have good pollen preservation. You can't have good pollen preservation. So you need to think, as I already said, of complementary proxies. So here, phytoliths may be useful. And it's also true that all pollen are not equally well preserved. So some may particularly not stand the chemical treatment and hence not be represented in the pollen spectrum. So these are some caveats which we need to keep in mind. But then I'll go on to telling you what are the features which we actually observe in the pollen. This is going to be really very quick. So one of the important features is size, of course, as you do with most microscopic objects. But then the pollen also contain apertures, which are called pores and which are also called colpi. So this round aperture is called a pore, and this elongated aperture is called a sulcus, and it's called, and the wall ornamentation itself can have some patterns like reticulation and striation. So what I'm going to do now is actually to show you how it looks, under the microscope. So this is a pollen grain of Sida, and when we look at it under the microscope, so I hope you can see the video playing. So basically the light microscopy is used in the same way that you use for sectioning plants. So we see a yellow pattern. So you start with the top of the grain, because within the microscopes, within the slide, this is actually a 3D object. So this is a pollen of Sida, and this one is one of Manilkara. So you see the patterns, and you see the exine ornamentation here. You can see the long colpus here, and you can see also the pore. And you can also see that there are actually 4 of them. So you know the symmetry, that it is what we call as 4 colporate. But this is what a fossil slide looks like. So the pictures, the videos I showed you, they were from our pollen reference collection, where we really want to know and study the morphology. But this is actually what this looks under 160 times magnification, and this is 400 times magnification. And here's one pollen which you can see. So it's not that great, but then normally we start training, and we train students by using pollen from honey, which is like really very rich and diverse. So you can see at least 10 different types of pollen grains over here. And this one is what we call as composite akinate, but it could be many things. It could be blumia, it could be sunflower, it could be a lot of things. And this, on the other hand, is belonging to Peltophorum, because that's quite a favorite of the bees. And then how do we collect the sediment samples? So this is just an illustration of one of the corers that we have been using. This is a modified Russian corer, and it's manually operated. And these are actually images from one of the field sites in Andhra where we have collected sediment cores. So the process is to collect the core, to analyze it, and finally to reach to this pollen diagram. And this again is one of the sites that that we had identified in the course of a project with the help of both remote sensing, because we had done this jointly with the NRSA at that time, and also the Forest Department of Andhra Pradesh, where we had a lot of help once we identified many sites like this. And this was the other site which we had identified. This one is Rushul Cheruvu in Mehboob Nagar district. The previous one is actually in Rajampet, and it's Ellamarazu Cheruvu. And then finally this one, Kotapuram Cheruvu, which is the one which I'm going to present the results of. This was identified in Lingal RF, and it was sample for pollen. And this is actually the location of the site. So this is Kotapuram Cheruvu, and we got a core out of this, and we also got it radiocarbon dated, and we found that we got a record which was going back from a 1 meter core, which was going back to nearly 1772 AD. So a little before that, probably. And this is what we call as the percentage pollen diagram. So now you see that I've marked a lot of taxa here, and you also see I've written Justicia T, which is a pollen type. So we also have a lot of the forest taxa. You have Hardwickia binata, which is one of the pollen which we can identify actually to the species level. But we also have a mixed group of Melastomataceae, Combretaceae. But in the dry deciduous forest, of course, this is an important component. We have Lania, we have Glaucidion, we also have Schleichera, as well as Dodonia. So what we did was also to quantify. So the other important thing with which allows pollen to be used in a very powerful way for past reconstruction is that you can quantify the pollen per gram of sediment, but you can also quantify using modern analogs what is the distance weighted pollen measure, and apply it using another modeling approach, which I will come to in the later part. But here, for example, what we saw with the pollen concentration diagram was that when we summed all the forest taxa, there was a slight increase on the top, which was a bit surprising. But you also see that one of the key disturbance markers, which is xanthium, has also increased quite a bit on the top. So which means in the more recent times. So we just took up this particular thing. So one of the conclusions of this work was that though the forest cover remained unchanged, some important species like this Hardwickia binata was not very evident. So even if on the top we had quite a good recovery, we still had quite a good recovery of the pollen types in the recent time. It was not. So we also noticed that there was this practice of loping of Hardwickia binata trees, and though it will not destroy the tree in the short term, it may affect flowering cycle and pollen productivity, and hence the entire reproductive cycle of the this particular plant. So we did this only with reference to this one species. And then we have published this already in Botany International. And then we continued a little bit more analysis looking at the rainfall data. So this is what I meant by saying that it's quantitative data. And we were getting some good results with reference to correlating it with the rainfall variability taken in different cycles of rain adjusted. So all of this is very important, but if we want to apply this pollen data into models, that is climate models, which are using feedbacks from land cover, then we need to go about in a slightly different way. So as I already mentioned in the caveats, there's no one-to-one relation between the pollen and the plant, and also it is only indirectly that the climate is linked with it. So we need mechanistic models to understand pollen vegetation relationships, and these have been developed in Europe and very well applied all over Europe and in many parts of some parts of North America and Canada, and also recently in Africa. So, and of course, my PhD student Navya did this work in Southeast India. So this requires us to do vegetation surveys, which is very, very important. And it needs to be distance weighted. So what do I mean by distance weighted? So you take a pollen sample here, and then you have around 10 meters, we have 21 quadrants of 1 by 1, because the assumption is that the vegetation closest to the pollen sampling makes the maximum contribution. And then we have a zone B, which is also surveyed on the field and by walking in transects and making these quadrants whenever there is a change in the vegetation. The last part, which is going up to 1 kilometer, it's done with remote sensing. So in addition to this field survey, which we do, and that's Navya there and also Prasad, we also need the fall speed of pollen. And all this goes into the ERV models, and it gives us an estimate which is called the relative pollen productivity. It also gives us the background pollen and the relevant source area of pollen. But interestingly, what we get out of this work, which is going to be applied to a fossil to the fossil reconstruction, is actually these 1 kilometer radius maps of the vegetation around each of the pollen sampling sites. So now we need to have, in order for us to get pollen productivity estimates for N taxa, we need to have 2N number of sites. And in her thesis, Navya has worked on 14 sites, and we have got the pollen productivity estimates for about 6 taxa out of that. So it's a minimum of 2N sites. So this is a important side output, because this actually gives us our sampling strategy makes us want to choose sites which are in different degrees of disturbance. So because of that, you get the chance with this to also map the vegetation in a systematic manner in various parts of Southeast India. So this was all done in and around Pondicherry mainly. So one output of this was this paper, which has been published 2 years back. But the other outputs, it's in Navya's thesis, and her papers will be coming out quite soon. So the whole rationale of this extended R-value model is that the pollen loading of a taxa in your sediment record, it's actually the distance weighted plant abundance within what we call as a relevant source area of pollen, which means that you also have a background pollen which can come in because of long distance transport of pollen, and you also have the relevant source area of pollen. So this ERV model, which allows us to get the relative pollen productivities, is then applied to what is called as a model which estimates the regional vegetation abundance from what we call as large sites. So why is all of this important? Because this is the data which actually links what is happening to the feedback from vegetation, which has now become very important in all the climate models. So as you know very well, it's the atmospheric composition and climate which decides what the terrestrial ecosystems are. But then you also have humans interacting with these ecosystems, and you also have the ecosystems themselves providing the cover and giving a feedback. So you have forcing because of the humans and also because of the climate, which is interlinked. And you also have the, again, you have feedbacks from the atmospheric system, which has been so modified back into the terrestrial ecosystems and so on and so forth. So one of the highest priorities of climate modeling community is to develop these dynamic vegetation models, and these have been done now. And the latest climate models are all incorporating the feedback from land cover, which is feedback from the actual vegetation on the earth, which had been ignored in previous editions of these models. And more recently, the pollen-based reconstructions themselves have been put into it, but of course, we are not yet there for the Indian part. But that's why this is very important, because this can be used in climate modeling, and it can be used also in conjunction with the information from land use, actual land use, which we need to interact with historians. Dr. Mathur had mentioned, so there is no escape with interacting with the social sciences and with people who are doing that. And history is really important, and we have also done that in the course of our work, and archaeology, because to understand the past, you need to know a little bit also from the humanities side, how they do the reconstruction. So now going back to the site where we have done the reconstruction in Southeast India. So this is a site which is called Chinabu Samudram. It's very close to, it's in Vilupuram district, and it's very close to Pondicherry. And here we got a 1.3 meter trench. We were not able to core here. And this trench dated back to nearly 6,000 years. So this was a very great thing. And we also got the pollen record, similar to the record that we got in Pottapuram Cheru, but we did all of this work of quantifying the distance-based pollen weightage and getting the relative pollen productivities for some of the taxa. And so we are able to go from the pollen data to the actual land cover. So what you see, for example, is that Acacia albizia is actually underrepresented in the record. So this is because of various reasons, because of its productivity, because of its dispersal, because of various other reasons. On the other hand, you see that Justicia is almost always overrepresented. And you also have another example where you have Amaranthesi, which is again underrepresented. So this is what we mean. So you actually get these values. These are actual estimates of the actual land cover, as opposed to just the pollen percentage diagram. So we use palynology as the central tool. We link with people doing geology and geochronology and archaeology, of course, but we also link with modern ecology, because recently we have also started looking at pollen loads from bees and how you can get the foraging preferences of these very important creatures. So we started doing it with honeybees because it's the easiest for us to start doing that, but it could also be done with other bees and other pollinators which actually gather pollen. And I just leave you with these two things. One is from Abhayar, which says, because it's a mixed audience, I'm not going to say the Tamil one, it's there. what we know is just a handful of earth, and what we don't know is as huge as this world. And for us, because we use something like the pollen to see a world in a grain of sand and heaven in a wildflower, and hold infinity in the palm of your hand and eternity in an arm. Thank you all very much for listening to me. And I'm most happy to answer any questions that you may have now or later. Thank you.
Thank you for that talk. Do we have any questions? Yeah. Maybe there's a mic right behind you. Oh no.
Oh yeah, okay. Actually, it's a simple kind of naive question about the method. So it looked like the depth was not, it was not a very deep core. So what happens when, people keep digging things all the time? So is there a possibility that, we could get mixed up with just human activity?
Yeah, that's right. You're quite right. When we choose a site, which is why actually we worked with, for choosing our pollen sampling sites, we worked with both remote sensing and also we used field surveys, and we used the knowledge which several archaeologists had about many of these sites already, because they knew that they were old enough. So we used a mix of things. We used very old topo sheets to know how old they were in the first place, because some of the water bodies can also be very recent. And it's true that you need to go to the, you need to check with the village maps, and you need to check what has been dredged and what has not been dredged. There is always some amount of disturbance. But when you put in all of these different criteria in place, then we in fact had a whole number of sites which were listed straight out of the remote sensing. But then we narrowed it down, and we only got to 3 of these places where we actually went. 2 of them we sampled, 1 we haven't. But we have a pretty good list still of maybe 10 or 15 where we'll have to go in, and we'll have to get the results. So normally we are pretty confident. The only thing we don't know is that the unknown is the sedimentation rate. So depending on the sedimentation rate, which can vary quite locally, you might have even a 4-meter core, which is dating only to a few hundred years or maybe just 1,000 years. But you may also have a 2-meter core, or a 1-meter core, which can date back even to 5,000 or 6,000 years. So I don't know if I can answer that question, but I mean, we can be in touch, and I can give you more details about that later.
Thank you. I have a question. If anybody else has a question, we could go. Or I have a question, maybe slightly naive, but do all species, do pollen of all species preserve equally over time? Or do you think there is a difference? Some pollen show up better than others in these records?
Can you repeat it, please? Sorry. You said, do all pollen
preserve equally well? Or do
No, they don't preserve equally well. In fact, we know we have large families of pollen which do not really preserve well, like Lauraceae.
Oh.
So we have also worked in the Nilgiris, where Lauraceae is a dominant taxa in the sholas, but we will never recover Lauraceae pollen from the sediment record. So
Right.
that is the reason why this modern analog method is really important. Doing surface samples and matching the pollen and vegetation and getting pollen markers of a particular forest type is a very important exercise. And in fact, we have done that for the Eastern Ghats. We have sampled at nearly 120 sites, and we have surface sampling, and it's going all the way from Orissa up to Tamil Nadu. So we have quite a good data set on that, thanks to our cooperation with NRSA and also the forest departments in all the places. We had quite good interaction with them, and that is how we actually do the vegetation reconstruction.
Okay. All right. Wow. Thank you.
I have a question, sorry. Are pollens a potential source of DNA? You know, they are very old, but have you tried extracting any genetic material from them?
Just 1 second. I want to switch my device. Can you please repeat?
Yeah. So I was asking if these pollens are a potential source of DNA.
Potential source of DNA. No, not the pollen in the sediments.
Okay.
Because the pollen in the sediments, they will be acted upon. You know, you have the soil chemistry acting on the pollen in the sediments, and all the cell contents will be exuded. Whereas the sediment itself, because the cell contents have been exuded, you can do ancient DNA on it.
Right. Thank you.
Are there any more questions? Yes, Dr. Parthasarathi.
Hello, Dr. Anupama. Parthasarathi here.
Hello,
Are
sir.
there any sites wherein the details of sedimentation rates quantified through some other departments like geology or earth sciences, wherein you could venture such studies?
Yes, indeed, we have done that. So we have done 2 cores in the Kaveri Delta, and this was done along with the Earth Science Department in Pondicherry University.
Okay.
And you can use grain size measurements to actually get the sedimentation rates. So, and you can do, in fact, yes, we have, and one of them is published in Catena, and the other one is being published in Quaternary International. So two locations in the Kaveri Delta, and those 2 cores have also been used in this reconstruction which we have done. It was not only for Chinabu Samudram, we have also done, and there also it went back to about the mid-Holocene, one of the cores, and the other one was about 4,000 years.
Thank you. I believe we have another question. Professor Mathur.
Anupam, this is Dr. Mathur here. I
Yeah.
must say that I did not completely hear your presentation. Might be you have covered, but since I have an opportunity now to say, see, the plant scientists and climate scientists are looking at phenology when we talk of detecting climate change.
Yes.
Now, do you think studying pollen in any way can help in detecting any change in climate? Is
Yes.
any kind of a correlation possible?
Yes, definitely. Because
And what is that, and who's supposed to study that?
Studying past pollen is definitely linked with climate. So one of the records which I had presented, it was a very short one though. It was only going back to about 200 years. But then we have tried to correlate it with the rainfall, taking the rainfall from the CRU data set. And we are actually able to get nearly 60% correlation of one of the pollen taxa. So there are methods now which allow you to do the correlation with climate. But more importantly, if we use this modern analog method, if we try to quantify the pollen-based land cover, which means that how much of the land was covered by these taxa in the past, then it has really good application in past models. And now past models are very important because past modeling is the only way in which you can check the models for the future is applying data like this, because pollen can directly go into that.
Thank you. One last question. Since this meeting had a special focus on red sanders, and since you've done work extensively across the Eastern Ghats, could you comment on whether red sanders is a species that whose pollen kind of saves well in the record? And if yes, did you come across any records of red sanders? Can you comment on the species in the past?
I'm afraid in the records that we have uncovered, we haven't really found the red sanders pollen. But I mean, it's always possible to go back and look at some of those cores, or maybe we did not sample in the right places. Like I said, we managed to do sediment cores only in 2 of the 3 places in Andhra, but there's a lot more. There are a lot more places where it could be done, and we have the sites. And I would be really happy, because now that you have the ICER in Tirupati, it would be really nice if you and your students can initiate such studies, and we would be very happy to give any help on our side which is possible. And I think it should be done, though.
That sounds really exciting. We're really looking forward to have you visit us so we can kind of discuss these things further and maybe look at what we should do next.
Okay.
Yeah. Thank you so much.
Thank you.
And we can move on to the next session. Thank you for accepting the invite and agreeing to do this, over Zoom. We really appreciate it. We were very keen to have you talk about palynology, to add it as a component to the work that we are trying to showcase. Thank you.
Thank you.
We now move on to the next session, which is plenary talks, and research talks, and Harsha Jagdish will do the introductions for this session.
Hi, everybody. We'll go on and move on forward for the day. Our first research talk is by Ms. Swati Udayaraj. She is a PhD. So our first talk is by Ms. Swati Udayaraj. She is a PhD student in IISER Tirupati.
Good morning, everyone. Today I'm going to talk about understanding patterns of squirrel occurrence in India, and then focus on to Tirupati. Giving a brief introduction about the squirrels. So based on lifestyle, we can divide squirrels into 3 broad categories: tree squirrels, flying squirrels, and ground squirrels. So most of the tree squirrels and ground squirrels are diurnal, whereas the flying squirrels are nocturnal. And with reference to India, we find tree squirrels and flying squirrels mostly in Western Ghats, the Deccan, Central India, and northeastern part of the country. Whereas ground squirrels like marmots are restricted to Jammu and Kashmir, Ladakh, parts of Sikkim, and Arunachal Pradesh. So now, why even study squirrels? So they are, first of all, a very understudied taxa, and there's lack of proper documentation of the same. And also, given that they are a good indicator species, it might be really interesting to study how the species respond to changes in the landscape. And also, given that in the same group, we have 3 different lifestyles, it might be interesting to see how they respond differentially to the same pressure. And also, these species tend to have high species richness in Asia, but are highly understudied, especially in this region. And now, why do we actually need occurrence data to begin with? So occurrence data helps us understand current species distribution as well as past. And this can help us understand multiple aspects of the species ecology, including range shifts. So given that traditional data sources like literature papers, museum data, and all do provide data, but given that some species are really underdocumented, we can utilize other secondary sources of data like citizen science and social media, which fill in the spatial gaps. Now, moving on to the type of data, what we are dealing with here. So the first set of data set is primary sources of data, which is also known as traditional sources. This mostly includes literature data, museum data, and also data collected internally from our lab with during multiple field visits. And moving on to the secondary sources of data, these are mostly opportunistic data collected by nature enthusiasts, and they have high potential to be used for scientific research, especially in areas where traditional data is lacking. So this mainly includes citizen science sources and social media sources. Now, so we collected and curated this database over the last couple of years. And for social media, we manually downloaded the images from multiple websites, and occurrence data for the traditional and citizen science data sources were also recorded. And each of these records were manually curated for species misidentification as well as location extraction. And total of 23,132 occurrence points for over 30 species were collected for Asia. And most of these species were records were found in social media, followed by traditional data and citizen science. So now our focus will be on the 30 species of squirrels which we find in India, and some of these species geographical range expands further in Southeast Asia, and so we'll be also including that part of this range in the study. So overall, if you look at, there are 14 tree squirrels and 14 flying squirrels and 2 ground dwelling squirrels in India. And if you, as expected, the tree squirrels seem to have over 20,000 record points, followed by flying squirrel and ground squirrel. So the ground squirrel, tree squirrel record points, where we see the traditional number, most of the contribution is from our lab. And as expected, social media seems to do better for all the three sources, all the three types of squirrels. So now, where are these records coming from? So we try to plot it on a map, and as expected, the hotspots have larger number of records. So if you look at the traditional source of data, most of it is from southern Western Ghats and central Western Ghats. Few are from central India, and some are mostly from the northeastern part of the country. And then citizen science also shows a similar image, but the number of records are really high from Kerala, especially due to high citizen science activity there. And similar image is also seen in social media, but we see a better representation of citizen science and social media data for the Eastern Ghats side. But if you want to understand this better, we need to zoom in a little further. And as you can see here, clearly that the Western Ghats has a really high number of records as opposed to Eastern Ghats. And this is in sync with what Dr. Mathur had explained yesterday, that Eastern Ghats lacks a lot of data for multiple taxa, and squirrel is one of them. So for traditional data, we are specifically focusing on the 4 states of Eastern Ghats, have not included Karnataka and Chhattisgarh in this analysis. So if you can see that even when we are looking at Tamil Nadu, a major chunk of the data actually comes from the Western Ghats side of it and not from the Eastern Ghats. But for citizen science and social media and traditional, as you can see, the data is more or less from Tirupati and Vijayawada side, which are actually our study site. So most of the data points contributed even here are from our lab. Now moving on, all these analysis, whatever I showed is mostly to do with these 4 species which are found in Eastern Ghats. So this is the grizzled giant squirrel, this is the Indian giant squirrel, the 3-striped palm squirrel, and the 5-striped palm squirrel. So moving on, this is how the record points actually look when we plot it on the Eastern Ghats side. So as you can see that a lot of these traditional data points are restricted to the Western Ghats side in Tamil Nadu and also some coastal cities. In Andhra Pradesh, as you can clearly see, a lot of these record points are from Tirupati, which is from a fieldwork, so as in Vijayawada area, and there is only few points in Telangana near Hyderabad. And Orissa has 0 record points for traditional data. So we move on to citizen science. It shows similar pattern, but there are few areas which are blank. And in social media, as you can see, the record points are much more as compared to the other 2 sources. And Orissa has really good social media points, but are all restricted to some areas. So you can clearly see that a lot of these points are actually coming from only certain areas, and they are spatially very, it does look like they are restricted to areas where there is higher human numbers. So now we wanted to understand whether, so we wanted to see how the records are differing across the states. And as you can clearly see that Tamil Nadu and Andhra has really good number of records, Tamil Nadu being the highest. And these are the 2 states where we are actually actively doing field work. And most of this data in this is from that. And Odisha and Telangana shockingly has really low data. And these two states really need to be prioritized in terms of documenting occurrence data. So now, given that in the previous two slides, I showed that there are certain patterns which we see that some records are only coming from certain locations. So we wanted to see whether there is any spatial bias. So we extracted values for human population density to see any existing bias. So this is the blue dots are the traditional data, the yellow ones are citizen science, and the green are social media. So over the years, for the last more than a century, you can clearly see that the traditional data decreases in numbers, but they were more or less spread across different areas. But when we zoom into the last 20 to 30 years, you can clearly see that a lot of these citizen science and social media have taken up after 2000. And this is a zoomed in picture of the second part. You can clearly see that though social media and citizen science both are biased, the social media is more biased towards area with higher human population density. So now there could be two reasons. Maybe there are more users in these areas, or maybe there are some species which actually are found more. So we looked at the species which are found, and we, as expected, we saw that the palm squirrels tend to be found more in urban areas and the records were high. So we wanted to investigate this further, whether to see the palm squirrels are opportunists in human dominated areas. So to understand this further, we used three-striped palm squirrel as a study system in Tirupati. And what we know about these species is they are opportunists in urban areas. They're also found in natural habitats like in forest or scrub lands, but they're possibly found in low numbers based on our previous observations. So they are highly commensal and stay in areas surrounding humans, and they're also being declared as vermin and pests in certain parts of the country. So, and also this makes an interesting system to study response to urbanization in small mammals, as they are found throughout the urban gradient. So now moving on to where we actually find them. So they are found in multiple types of habitats, which includes scrublands, grassland, forest edge, plantation, agricultural and urban areas. And even though they are found in multiple places, the numbers are not more or less same. So there, it looks like they might be doing better in the urban city centers. And this is something we want to test with the data. So, and also multiple factors like food resource availability, tree cover, predator presence, and etc. might have a role to play in determining its abundance. So these are the potential scenarios which we had hypothesized. And so we either the numbers might decrease or it might increase as we move across an urban gradient, or it might increase till a point and plateau out. So so this is the study site where we had actually carried out this occupancy study. So if you look at this is Tirupati, it's in South Central Western Ghats. So it's a perfect natural laboratory to answer this question, because mostly this landscape is surrounded by SV National Park on one side, rural areas on one side, and also there is a mosaic of urban settlements. So basically there is this study site has highly heterogeneous land use cover, and this gives us a very good opportunity to understand how the same species is faring in all these different areas. So now this is how typically Tirupati looks like. You have agricultural lands, then you have urban settlements right next to a national park. So there's a perfect gradient for us to understand how species might fare in this area. So now to address the same, we divided the study area into 4 broad categories. So this was based on human presence. So this orange area is the urban area. The yellow one is the intermediate area where the urbanness is a little less. And then we have the blue rural areas and the green uninhabited or forested area. So some of these are forests and some of these are scrublands with no human habitation. And so based on a power analysis, we decided to do 317 grids across all these categories. So these are the grids which we did. So the small dots as you see are 100 by 100 meter grids. This was based on the squirrel home range size. And this is the data we had collected last year. I'm going to present the same. So basically we had few variables in mind when we went out to collect field data. And for this talk, I'll be specifically focusing on the anthropogenic and the invasive aspect of the same. So the plus sign indicates that we expect the variable to impact the squirrel occupancy positively and the negative ones negatively. So now moving on, so we saw that as expected, the tree density is positively affecting the probability of palm squirrel's occupancy. And as expected, as the human population density increases, so does the probability of occupancy of squirrel. And this is in sync with what we had hypothesized in the earlier stage. And also, interestingly, we saw that there's a pattern with Prosopis julifera, which is an invasive species, and we have seen the squirrel actively feed on this on certain instances. And there seems to be an increase, but we need to collect more vegetation data, which we'll be doing this year to understand this pattern better. And to look at how the detection probability is changing across categories, we can see that the urban area has high detection probability as opposed to intermediate, and rural has very low. And also in case of occupancy probability, somewhat similar across these two categories, but very low in rural areas. And just to look at squirrel sightings on trees, so we didn't get any pattern for ornamental trees, but we still have to collect few more data. But just to look at some rough pattern about where we are seeing the squirrels more. So most of the squirrels were found in Peltophorum pterocarpum, which is an exotic flowering tree. And also in some of these fruiting trees, which are planted by humans, and they are very less found in native trees. And also in few instances, we have seen them on red sanders in few of the grids. So these are the ones which are positively affecting its occupancy in the landscape. So now this is in sync with what we had got. So further more data has to be collected to understand whether it's the orange one or the green one, which is being followed here. And if you look at the study area, the lighter white shades is actually the areas where we have got more squirrel presence. And this is showing that it's more or less present more in urban areas as opposed to surrounding areas, which has less human presence. So moving on to summarize the talk. So Eastern Ghats has very low squirrel records, and secondary data sources are something which are filling in some gaps, but we still need a lot of rigorous surveys to understand what the species distribution is in the landscape. And we can use citizen science and social media as a good tool with more public participation. And also this can be encouraged with targeted drives. And species like three-striped palm squirrels are doing better in areas with high human presence. And with further studies, we can understand how they are surviving in these concrete jungles. And they tend to have a preference for ornamental and invasive trees, and which is actually an indicator for their suitability in disturbed habitats. And future work, so we are creating this Squirrels of India database, and we'll be making it for open for public use. And so this is also this is the first database for squirrels in India. And this data will help us understand patterns of species distribution over the years, and also create biodiversity maps, which can be further utilized to prioritize research and conservation on sensitive taxa. And also we'll be giving out details about how we went about this so that people can utilize this as a template for further studies of unknown taxa. So yeah, that's it. Any questions?
Yeah, so I have a question about your observation of the red sanders connection. So do you think there's any kind of ecosystem service that is there with the squirrels? Like, I mean, squirrels are known to disperse seeds.
So they are pollinators. They are seed dispersals in most of the cases, because squirrels mostly feed on fruits and also sometimes on flower nectar. So they visit multiple flowers. So they can be pollen transfer. There could be roles of pollen transfer, but they are actively known to transfer seeds from multiple places. So that is why one of the reasons, like the invasives might be spreading more, because a lot of these invasive trees which we see in this landscape have lots of fruit, they're fruit bearing. So there is a potential chance of them being spread, like the same case for bulbuls. So something like that.
So you are telling that from citizen science and social media, you are getting more data.
Yeah.
What about from forest department side?
So forest department side, so most of these data is traditional. Basically, since it's collected in a very rigorous way with proper location, we are clubbing that with traditional data. So most of this data is out there in literature or any Zoological Society of India book and all. So all this is being clubbed in traditional. Citizen science and social media doesn't, it only accounts for data which is taken by general public who are not researchers, because they are not really well versed with like how to take locations accurately, because these two data sets require more curation, and traditional data is actually much more reliable. So like you said, forest department data all comes under traditional data set. So a lot of those has been included in this from multiple
Do you
databases.
mean to see that they say that the data given by the forest department is more valuable for your study purpose rather than citizen science and social media?
So with respect to spatial accuracy, more valuable, but with respect to numbers, I think the citizen science and social media might be much better because the numbers are large. That's the thing. But with respect to spatial accuracy and other aspect, forest department data will be better.
Now coming to especially my area, what is your observation?
So in this landscape, we have mostly worked in urban sites. We have not done much work in the forest landscape. So I won't be able to tell much about the undisturbed areas, but we are seeing that the numbers are high in urban spaces. But yeah, so that's the observation we have as of now.
Okay.
Yeah.
Well, my compliments first for a very nice study. You did say that squirrels are basically trying to tell us what urbanization can lead to.
Yeah.
But when you also said that there are 4 different species. So which ones are you talking are more affected by urbanization?
So so this particular occupancy study in Tirupati, you only find the 3-striped palm squirrel. So
Okay.
the 4 species I actually have shown analysis for all over the eastern Ghats. So basically, grizzle giant squirrel, you find it only in Tamil Nadu and some parts of Karnataka. Just a second. Yeah, so this species is not found here. This
Okay.
is just for the tree squirrel analysis for all over the eastern Ghats. So the giant squirrel you find in Tirumala and other forested areas surrounding Tirupati, but not in our study area, which is more or less centered towards urban and human dominated landscape. And this is the species which we are doing the whole occupancy study on.
Okay, fine. See, you also understand the concept of surrogacy. So can this species also mimic any other species whose status we want to know in an urban environment?
Yeah, so a lot of the urban exploiters, like let's say pigeons, crows, they tend to have similar characteristics. Like in general, there are a lot of meta-analysis which looks at particular taxa. So overall, for mammals, at least it's known that larger litter size tends to be very crucial in determining whether they tend to do well in urban areas or not. And the other important aspect is their flexibility with the resources they use. So the more omnivorous they are, or the more adaptable they are to changing areas. Like for example, in one of our studies, we have seen that palm squirrel tends to use a lot of these anthropogenic materials for nesting. So the more flexible they are, the more chances of them being really successful in urban areas. Like for example, if you look at giant squirrel, these 2 species, their ecology itself is very different. They require fruiting trees, they require high canopy cover, which is actually lacking in an urban city. So these species might not be very well supported as opposed to these 2. So even the species ecology itself will determine whether they are fit for the place or not. So there is multiple factors which actually determine whether a species will fit the bill in like surviving in an urban area.
And most of these are observational studies, or you also count nest and anything
So
else?
we do take into account where we see it, but here we are not monitoring nest. So here all we are doing is conducting occupancy surveys, taking data and habitat data, and also doing vegetation plots.
Do you think that would be the next step? Because we would need some rigid baselines for long-term.
Yeah, that's something we are planning to do, but we are not yet started. So this is one of our long-term sites. So we do plan to do a lot more work other than just field surveys.
Perfect.
Swati, you were mentioning about indicator species. So partly, some part of the question Professor Mathur asked met with. One is about the usage of the word indicator species. What do they really indicate?
So basically, these 2, at least the palm squirrels of this Funambulus palmarum and Funambulus pinnati in general, they are found in very human-dominated areas. So somewhat it can be used as a proxy to know whether the area is disturbed or not. So they tend to do very well in disturbed areas as opposed to natural areas. So hence they might indicate what kind of a place we are dealing with. And also these 2 species in general tend to do really well in highly vegetated place with tall trees. So again, this also shows an indication of what kind of vegetation is there in that place. So vegetation, if it's gone, these species are likely to vanish. So this is something which will help us understand what is happening in that place. And over the years, if we see the numbers are dwindling and coming down, it also reflects the vegetation profile of that place and how it has changed over the time.
Okay. You're using the phrase internal lab data. It can better be used as present field data. Because you have
Okay.
collected it.
Yeah. So
And-
as part of, so that was for the database. So it was not just this field, it was also multiple field sites across India. So that is why I use that term. Yeah, I'll keep that in mind and change it.
Better word,
Okay, sure,
because
sure.
you are writing thesis.
Yeah, sure.
Other is, Robin was talking about indicators, ecosystem services. Ecologically, the squirrels are better labeled as seed predators and accidental dispersers in the process, number one. The second part is, what is the extent to which they have the habit of storing seeds in tree holes, and to what extent across these types and species such trait remains?
So we know that for giant squirrel for sure, but I think so most of my observations for palm squirrels have been limited to urban areas, and they have been like more or less been feeding on human subsidized food. So and in general, but they are known to like transport it from one place to another while they forage on the same. So distance wise, I'm not sure how far it can go, but if that's your question, that's not something I really know well about the seed dispersal properties.
Okay. Your database is a welcome move. So good effort, that could be valuable. And yeah, you can have more sample in forests.
Yeah, so I have only sampled in 3 categories. The other one we have just only done partial sampling. So that's why I didn't present it in this. So we do have a lot of samples. So we will be finishing the field work by June. So by the end of it, we'll be having data for the same.
And the other is, of course, monitoring. So the future efforts, that could be valuable.
Yeah.
Okay, thank you.
Thank you.
All the best for thesising.
Thank you.
Hello. So Dr. Anupama has pointed out that in urban and rural areas, it will be good to know the typology of housing. Sorry, was I audible before? I'll repeat it. So Dr. Anupama has pointed out that in urban and rural areas, it would be good to know the typology of housing that squirrels prefer.
Yeah, so we are not like actively monitoring nests now, but wherever we are seeing squirrel, we are taking into account the immediate surrounding. So none of the data has been like added in this. So since in the scope of this talk, I won't be able to present all that. So we do see some patterns, but that will be like discussed at some point. So we do see there's a different type of housing categories, or even within houses, there are different like apartments and individual houses. So we have data for all that, but that's not been presented here.
So there was one more question. How is species ID from citizen science tools verified and used for research analysis, especially in the case of flying squirrels, as morphology and ranges are hugely understudied?
Yeah, so we have a key species identification key, as well as we have a rough idea about what the geographic range is. So usually a lot of these citizen science and social media data are only used if it is associated with an image. So if there is no image, we completely discard it because that's something not reliable and we won't be able to verify it. And it's usually matched with that ID, and we take a consensus of a few lab members who are actively curating that. And after that only it is taken. And if there is any confusion with what it is, we completely discard it without actually considering it for further analysis.
In the food chain, where does it occur? Is it what are the predator of this animal?
So there are lots of few predators even in the city, like dogs, cats, shikras. There are few raptors here, and we have seen them being actively like attacked by these species. Shikra is a small sized raptor. It's almost like a little bigger than crow. It's an accipiter group. So it's one of the very commonly found birds in the. Yeah, that is also one of the, but the most like largely found in this landscape is at least shikra. So there are other like one black kite and other raptors in general tend to feed on squirrels. Sir, I'll show you a photo after this. Hello.
Yeah, maybe we should wrap up and move to the next talk. Swati can take questions over the break, tea break, but if you want to stick to time. But just a quick response, the lab has studied nesting patterns of squirrels, just not in Tirupati, just not part of this work, quite extensively in the Western Ghats. And the concept of squirrels being seed predators is largely outdated. It is now we know that they play many more roles in forest ecosystems. So,
Thank you.
Thank you, Swati. Our next speaker is Dr. Shivani Jadeja. She is the Assistant Professor of Biological Science from Priya University. I welcome Dr. Shivani Jadeja.
Am I audible at the back? Am I audible at the back? Thank you so much. Well, I thank all the organizers for this opportunity, especially Dr. Nandini Rajmani, for me to be able to be at this platform and just share some of my work. I'll admit right at the start, it's very preliminary, more or less exploratory work at this point. But I'd like to use this as a case to, I don't think I'll top what Swati's done before, but a case to sort of highlight the strength of citizen science, to understand, in this case, spatiotemporal patterns of occurrence of Dna butterflies. And I don't need to convince anyone in this audience that we need to know where organisms are to be able to understand their ecology and know whether conservation measures are needed, and if so, what. And animals move, some migrate, and so protection in certain parts of their range may not be sufficient. And even those that are residential may shift their ranges over time. I'm going to highlight with this one example of monarch butterflies in the US, and various forms of science over 100 years, including citizen science over decades, have allowed them to come up with maps like these to understand where the monarch butterflies are migrating and where they are, sites where they are wintering. And not only that, owing to citizen science, over time, they've been able to find that their populations are declining, and urban populations, migratory populations are declining and urban populations are increasing. And they're seeing shifts from populations moving from being migratory to residential, partly because of increased, possibly because of increased availability in host plants in urban spaces, in urban gardens. And they've been able to do this because they have all that baseline data. In contrast, in India, we also have migratory Dna butterflies, and they migrate differently. They migrate longitudinally, and instead of in response to temperature, they tend to migrate in response to moisture rainfall. But we have only opportunistic observations that have resulted in a model like this from Kunte and his group's work, Krishnamurti Kunte, which shows that basically in the pre-monsoon time, they migrate from Western Ghats to the Eastern Ghats and plains, and post-monsoon, they migrate back to the Western Ghats. Again, this is based only on opportunistic data, and what we tried to do in this work involves the work of Naveen Prasad Alex, who's an undergraduate capstone student and a project assistant who's worked with me for over a year as a project assistant. Here, this is from literature. We tried to see what do we know about the migration from literature. Each arrow shows the direction of migration from surveys, and the location are shown. And I'm only focusing on these areas in southern India over here. And you can clearly see there are just many more surveys from the Western Ghats areas and fewer in the Eastern Ghats. If you look at the colors, they match they've been coded to match the previous model I showed you. And you can see the pattern matches where mostly the pre-monsoon, you see migration towards the Eastern Ghats and plains, and post-monsoon, shown by the red arrows, shows migration towards the Western Ghats. Now, again, this is very patchy, and there are probably many other surveys going on, but this is what we could access online, right? What we wanted to do was see whether citizen science can play a role in filling gaps in our understanding of their occurrence and movement. This is just to show the kind of occurrence that exists for 18 species of these Dna milkweed butterflies in southern India, and this is from the iNaturalist platform that from where we've used the data for what I'm going to share today. and just addressing two basic questions today: whether we can understand patterns of occurrence and movement from this citizen science data, and whether what we see matches what is available through literature in terms of models of how these butterflies are moving. This was done with 5 species of Dna butterflies: common crow, blue tiger, dark blue tiger, plain tiger tends to be residential and more widespread, and common tiger. This was, we used only observations that are considered research grade. We went through all the images to verify it's the right species, it's an adult individual. And we only used observations from 2010 to 2021, because before that, the citizen science observations within a year were too few. This is a histogram that shows on the x-axis, it shows the year, and y-axis, it shows the number of observations for each of the different species across the years. And as we've seen in many citizen science studies, we're seeing more observations in recent years. So with this, and this is again only data from 2010 to 2021 in southern India. As I said again, we wanted to see whether we can use this to understand how species are occurring and how they're moving across seasons. So again, we classified the seasons based on the model that we had from literature. Here, there's just for providing a guide, there's a line that goes through Kanyakumari, horizontal longitudinal line, that vertical line that goes. And this is the pre-monsoon, all the yellow points, and you can see they tend to be more in the Western Ghats for many of the species, except for, say, the plain tiger, which we know is more widespread. And we do see still observations from Eastern Ghats and plains areas. If we go to the next season, this is the monsoon season. So if we look at the yellow arrow below, the expectation is that from during pre-monsoon, they move to the west, Eastern Ghats and plains. And so you'd expect more points to be on the right-hand side of these maps. So I'm just going to go back and forth a couple of times to show you that we do see this happening from pre-monsoon to monsoon. You see more occurrence points on the Eastern Ghats and plains side. So it tends to match what the explanation that's there, but it's still, as you'll see in a bit, that it's not as clean as we'd like to see. Then we look at post-monsoon. That's when you'd expect them to move back to the Western Ghats. And so if I go from there to the next season, you should see points moving more towards the left, which is towards the Western Ghats side, which is what I'm going to go back and forth, going from the red, which is you see more points going back, but it's not that strong for all the species. Another way to see the same thing is because we're focusing on longitudinal migration. Here on the x-axis, there's longitude. And again, you have the dashed line, which passes through Kanyakumari. And the y-axis shows the proportion of observations. So as there are more observations on the right side of the dashed line, you are expecting to see more occurrence records on the Eastern Ghats and plains sites. And where you see a strong pattern of migration that matches the model is in the case of the dark blue tiger, where you see shift on this side and then a strong shift towards the Western Ghats. But in many other cases, in most cases, you still continue to see occurrences in the Eastern Ghats and plains throughout the year. And so just with this very, very exploratory work so far, two main points. Yes, patterns of migration broadly match the models we see from literature, but it's not that simple. We see occurrence records from Eastern Ghats and plains throughout the seasons, and that begs more research and more analysis. So I'm going to sort of end with talking about where our research, we hope our research would go, is first at least do more analysis with the data. One can sit and debate about which months are monsoon for Eastern Ghats versus Western Ghats, and maybe focus at a different scale of time. When more data becomes available, which is becoming a little tricky right now, it would become possible to look at changes over years, whether or not they are there. And also one of the things that we're working on is to correlate these occurrence patterns with environmental variables. Again, these data are available through various databases. We're using Copernicus database. I'm not sharing those results here. It's ongoing work, but we can then correlate what we see, the patterns over time and space, with temperature, rainfall, and such environmental variables. And finally, in isolation, these citizen science projects would not be strong enough. So the hope is that we would be able to follow up these occurrence records and patterns with field studies and citizen science initiatives on ground that involve public engagement that can then be similar in some sense, and maybe even more context specific in the Indian context, but similar to the kind of records we have for monarch migration in the West. With that, I'd like to thank the funding sources that supported this work, and also some of the data was collected with the help of undergraduate research interns, Aditi Balaji and Meghna Rao. And if there are any questions now or later, I'd be happy to take them, and look forward to feedback and discussions. Thank you.
Thank you. Do we have any questions?
It's not very clear. So it's believed to be in some places. It's believed to be. I'm so sorry. The question was about what's the lifespan. It's not very clear. It's some places believed to be some months, less than a year. I'm still trying to explore, and maybe I need to work with more lepidopterists to figure out. But there are questions of whether they migrate one site and then they die, or they come back. And there's some people report anecdotally that sometimes the ones that come back, say, to the Western Ghats, their wings are very highly damaged. And so there are questions of whether they might survive or not, or whether they, once they come on the Eastern Ghats side where they breed. Yes, absolutely, absolutely. So with tagging, that is something that would become known. Thank you.
The lifespan is only 1 month or something like that. Tagging will not help.
It would if we, for example, have collaborators both on the Eastern Ghats and Western Ghats side, right? Because then when they migrate, then citizen science initiatives on the other side can capture, and then one can see based on the kind of damage as to maybe, or based on tagging, yes.
What you're observing is only the butterfly stage, not the larva or pupal stage.
No, adult,
So
yes.
that will be quite small.
Yes.
Hi, it's an interesting observation. It's an interesting observation that you found some species that occur through the year in some parts of the Eastern Ghats. Where are these places? Like, are they climatically very different, or what's happening?
I haven't gotten into those analyses yet. So at this point, it's very exploratory. But since it is citizen science work, we can expect it to be only certain urban, peri-urban, rural areas. And this is where, this has to be paired up with field-based work so that we can get sites where people will generally not be traveling, let's say in protected areas, and get information from there. But there are, if you look at the maps, they are both sites in the Eastern Ghats and plain areas. So very variable environments. What is not clear from what we can't say from this is whether, is how long they spend there, right? It's just an occurrence record. And so it could just be species, individuals moving from a site, and this got recorded on the way. And so that is something we won't be able to say what it means. One of the, it's a plan, it's a thought that down the line, once there's some more baseline data, then we can work with those who, there are so many people interested in butterflies, and then people in local communities, they can come together and engage in a tagging program, in a monitoring program. This is quite common in many countries, and they've got decades of data through that, and they've been able to assess change in species behaviors, life history strategies, like shifting from migratory to resident through these citizen science initiatives. So basically, people who are not trained and are not doing this for a job get involved in this because it is something that they begin to care about, and it's around them. Just like this, there's a lot of work like that with birds that's happening increasingly in India. Less so, there are initiatives, but less so with butterflies. I'm looking forward to a lot of talks later today that do look at other citizen science initiatives that are going on.
Hi, I had a question.
Sure.
So these butterflies, which you're saying that usually they were only recorded in certain seasons, but now you're getting records from all across the seasons, right?
I wouldn't put it that way. The thing is that the model that's there is based on opportunistic data.
Okay.
It's based on surveys on 2-3 sites, and then directions from that are being used to extrapolate what the pattern of migration is going to be. So we don't have the detailed information. In fact, we don't have detailed information on where so in the Western Ghats, they form congregations, a lot of these butterflies. they'll form congregations of thousands of butterflies in a short space. And so there it's easy to quantify, but when they move to the Eastern Ghats and plain sides, we don't see reports of such large congregations. So we have very little understanding of what habitats are important and are they in protected areas, what elevations they are. And we need whatever information there is based on 1 or 2 studies, 1 or 2 sites, very small sites.
So this is largely like user-based bias rather than the species not coming there or something like that, right?
Yeah, it's just we don't have baseline information yet.
Okay.
And that's
So I had
goal.
one more
Yeah.
question. So do these species have any invasive host plants, and whether if their spread might inhibit them from going back, like they might not want to go? I'm just like curious. I don't know much, so I'm just asking if they have any affinity to it, and if there's increase in that, is it a possibility that there might be some effect in their migration pattern for the same?
I'm not aware of specific examples of host plants, but nectar plants, definitely. Lantana is one example that I can think of off the top of my head. But with host plants, they tend to prefer milkweed plants. And with each of the species that I talked about tend to have higher preference for specific milkweed plants. And I'm not sure if there's a pattern with any invasives or in that realm.
Okay. Thank you.
Thanks.
To everybody. Thank you. Do we have any other questions? Maybe we can have it later. Thank you. The next speaker is Mr. Harsha Kumar. He's a PhD student at ISA Tirupati. I invite him for the talk.
Hi, this is Harsha, and today I'll talk about the influence of citizen science drives in documenting moth diversity of India. And this work was done by Harsha Kumar, that's me, Senan D'Souza, Suyash Sawant, Viral Joshi, and Nandini Rajamani, who are all here. The status of moth documentation in India has had a patchy history. There are about 10,000 estimated species in India, and only 2,000 species have been documented post-2005 by citizen science efforts. And really the only comprehensive work on moths of India dates back to the 18th century. And this is work done by Hampson, a Britisher at that, who lived here at that time. And this is possibly the only information that's available for Eastern Ghats at the moment. And we don't have answers to simple questions like what species are here, what are their distributions, do we know anything about their early stages? And at this point, we need baseline natural history data that needs to be collected before we can address any real research questions. So citizen science events not only generate awareness, but also contribute to documentation of biodiversity. And the National Moth Week is a global citizen science event that happens every year in July, which is exactly aimed at doing this. It's a week-long event that happens every year. And it has kicked off only in 2017 in India. So with this, we ask if citizen science platforms are known to contribute significant data, but do targeted citizen science events like these National Moth Weeks accelerate this process of data collection? And we ask if they cause an increase in the number of observations or uploads. Observations are happening in real time, uploads in the sense that somebody could click a photo today and upload it at a much later point in time. And OTUs are proxies for number of species because of how iNaturalist deals with identification of taxon. And we ask if these events cause an increase in number of participants. This is like a community building exercise. And how many people contribute to this data? Is everybody contributing equally, or are there some who are contributing way more than others? And where are these observations coming from? Are they everywhere in India, or are there some clusters and hotspots and cold spots? And we end with a display of interesting findings at Tirupati, which were made during the Moth Week 2020. So we collected data from the Indian Moths project on iNaturalist for 4 years, and we downloaded data in 10-day time bands, which is approximately the duration of the Moth Week. And we use operational taxonomic units as a proxy for number of species. So, there might be many species that are identified only at the genera level, but they're all pooled together and put down as one unit because there's no way you can tell them apart. So this is a conservative way of saying how many species there might be. And we look at heat maps of activity generated on ArcGIS to visualize the geographic spread of where these observations are coming from. So to begin with, let's ask if number of observations or uploads so do the moth weeks affect these? So here you can see, okay. In the first pie chart, you see that the national moth week observations correspond to about only 16% of the data, of the total data. And there's about 83% of the entire data is coming from non-moth weeks, from other times of the year. And if you look at number of uploads, it seems like a lot of people are uploading older observations during the moth weeks. And you see like a significantly larger proportion of uploads coming up. And this is showing you the same thing, only here you have 10-day time bands here. And the year is shown in the bar. And each gray cube is the position where the national moth week happened. And here you can see that everywhere there's like this square, wherever there's the moth week happening, you see like an increase in the number of observations and uploads. But only in the year 2020, you see that there has been like a significant dramatic increase in both the number of observations and uploads, with the number of uploads greatly exceeding the number of observations. And this could be because the National Moth Week 2020 was incentivized. They said, if you post 500 records or more, no matter when you click something, you'd get a free t-shirt. And that's the one I'm wearing right now. So now we can ask if the moth weeks also cause an increase in the number of species that are being observed. And again, you see very similar results. You see that the number of species that are being observed are very, very high during the moth week. And this corresponds to the number of observations. And now you can see this is a nonlinear relationship. And it's indeed true that larger number of observations translate to larger number of species being observed in time. Okay, so now what's happening to the participants? So again, you see that the participants are also increasing during each moth week, but they fall off after the moth week has happened. So we're losing people. Something is happening here. And there are more uploaders during the moth week than other times of the year. So again, you see that the number of uploaders are very, very high when compared to the number of observers, which means that some people are only uploading old records and not watching moths during the month. And I know several friends who have been doing this. And now you can ask if the number of observations are dependent on the number of users, right? And here you see, this is a way of asking if is everybody contributing to the data equally or not. And here you see that the number of observers are not indicative of the number of observations made in each time step. And this is to suggest that the data might be highly skewed. Some people might be contributing way more than others. And here you see that 95% of the contributors have 40 observations or less on iNaturalist. And that 40% of the data that's on iNaturalist right now is contributed only by 20 people. And let's say you filter everybody who has 40 observations or less, and yet again, you recover the same patterns. So do moth weeks change anything? Does everybody have an insane effort during the moth week, or what's happening there? Again, you're recovering the same things. The same people are actively going out and looking for moths even during the moth weeks. And here you see 2017, there were 87 observations and 4 people contributed to 50% of the data. And here again, 5 people in 2018, 7 people in 2019, and 7 people in 2020, and 3 of us are here, the 3 of the 7, right? And the pattern's growing stronger every year, and there are these power users who are contributing to this data and skewing the average. So how are the observations spread out across the moth weeks? And you can see here that this scale bar here is a heat map, and the blue colors indicate low number of observations, and red indicates high number of observations. And you can see that the number of spots are increasing through time, which means that number of locations being sampled are increasing with time. And Bangalore was a hotspot in 2019. There are some parts of Kerala which were hotspots in 2018 and 2017 when the event just kicked off. And there's a spot in Mumbai in 2020, and the spot in Tirupati, which was consistent in 2020 and 2021. And those are mostly efforts from our lab. And you can see that most of Andhra Pradesh otherwise is empty, right? And many parts of central India and North India have little to no observations across the years. For example, Bihar, UP, and Madhya Pradesh have very few observations. So these are potential places where citizen science efforts can be targeted in the future to have like an increased geographic spread. Okay, so now we can ask, why is this happening? Like, why is it so patchy? Like, why are we not getting observations from everywhere? Is there a reason? And to do that, we compared the National Moth Week 2020 with another citizen science event, the Great Backyard Bird Count 2020. So the idea behind this is that there are naturalists only in some places, and the people who are watching birds are probably also the same people who are watching the moths. And yet again, you see that there's like this very strong parallel. You see the exact same thing that's happening. Like it's an exact mirror of the patterns that you see. And this might have to do with the strong foothold and growing communities of naturalists across different locations in India. And there is no easy way to address this. Okay, so if you look at all Moth Week observations, that's about 45,000 observations, you see that there are so many locations that are now sampled. And there's like this slow trickle effect that's leading to massive documentation of biodiversity. While observations are spread out across the country, there seem to be like a prominent cluster. You see like a lot of greens and oranges along the Western Ghats. And there's one in Bangalore, and then there's one in Chennai and Tirupati. And there are other like Assam, Tripura, and there are other small spots as well that you see that are probably growing. Okay, so here we have some interesting observations that we made during Moth Week 2020, which are very, very significant. Here's a Southeast Asian species, and this is the first record for India. Here's a few first records for the Eastern Ghats, first record for peninsular India of a moth that was first seen only in Northeast India. And here are two extremely rare cases where this is the first digital photographic record of a moth, because they were described in the 18th century by Hampsons in Tranquebar, and they were never seen after that. So this is like the first photographic evidence for these two species of moths. And here you have another first record for peninsular India, first record from Eastern Ghats of what is an African species. And here there's a Western Ghats species showing up in the Eastern Ghats. So to sum up, a total of 386 taxa were identified till the species level with a fair degree of certainty, and several more were left at the genera level. And by morphology, it looks like the numbers are going to reach the 500 mark. And all this is from my balcony at Tirupati. So the targeted events like the National Moth Week help document biodiversity. Incentivizing citizen science drives probably encourages healthy competition and participation. Though like only a few users are contributing to most of the data, there could be scope for community building exercises where you could set up an open moth screen. Like I could set up a moth screen and invite people to come look at my moth screen, right? And this is something that's taking over very quickly in India at the moment. And observations are trickling in from across India, but there are definitely hotspots. Now, the real question is, will you set up a moth screen at the next event? And it could be as simple as switching on your tube light in your balcony. And this is what I've been doing. Yeah, with that, I'd like to end. And this is where I live. This is where I did all the moth observations.
Yeah, Harsha, first off, I mean, that's a fantastic talk, and really amazing to see, you and others put together so much information. That's just brilliant. And, as a way forward, I was wondering, if, like you say, there are a few power users and there has to be this trickling of capacity building through the community. So probably these power users, I mean, we need to find a way to work with forest departments and other colleges and institutes, maybe even IITs and other places. How do we get others interested So maybe like stepping out of the balcony and to some other public spaces, like maybe have something at Divya Ramam Park or something like that as a event. So something that can encourage other people to kind of join in some of these events. I mean, I don't know, it's for you guys to think about, but overall, fantastic talk. Thanks.
Harsha, you told that moth population is not documented well. Agriculture crops. So it's all very well documented. I don't know much about the forest entomology, but at least in agriculture sector, agriculture entomology, so most of the moths are recorded. It is there, so you can consult. So any agriculture research station.
Correct. I agree with what you. They either fruit piercing moths or they either moths of the family Crambidae.
I know. So during my BSc agriculture study, I was knowing more than 800 species by every crop. So there are huge number of pests, large number of pests. So invariably, they're all moths only. As far as I know, there is only one butterfly pest, that is on the pomegranate butterfly. Otherwise, 80% of the damage caused to agriculture crops, both horticulture and field crops, by moths only.
I see. All right.
So,
I'll have a look.
So you said that the data is lacking. I think it's perfectly documented. So you can consult any system of agriculture, any institute engaged in agriculture research. Tirupati, there is agriculture college. Thank you. That is maybe one input. But we have a museum in Srishalam, and we had one here also in Tirupati. I don't know whether you can have a look at it. And we have one more in Kakinada on the Agri Foundation from Forest Department side. There's a good documentation of moths and butterflies. We're talking about moths. So maybe you have to widen the scope beyond the balcony.
Sure.
Other question? Thank you. Thank you, Harsha. We have our next speaker today, Ms. Rutuja Kokade. She's a PhD student from Kriya University and will be joining us online. I invite Ms. Rajita.
Please start your presentation.
Yeah. Good morning. Thank you so much for having us. My name is Rutuja Rokde, and I am an undergraduate student in biology at Kriya. This paper emerged from my research for my capstone project mentored by Dr. John Matthew, who also co-authors this paper with me. Next slide, please. I'm going to start with the Cytrodactylus Jaipurensis, commonly known as the Jaipur Bento gecko, named after Jaipur town in the state of Orissa. But Jaipur and the area in and around it is known not just for biodiversity that has cuddle value, but also for biodiversity that is not often rated. And we are looking at that which concerns public health. Next slide, please. And here is an example of another animal that bears the specific epithet of Jaipur, and that is the Anopheles Jaipurensis. Next slide, please. This particular talk is going to focus on the Anophelines of Jaipur in Orissa rather than the more commonly known Rajputanas Jaipur. And it is germane to this particular seminar because it is located in the Eastern Ghats, part of Orissa, abutting the present day Andhra Pradesh, and not too far from Visakhapatnam. Next slide, please. This particular talk places public health front and center through the work of colonial officer in 1913 called Edmund Ludlow Perry, born in 28 June 1872. His father was a Surgeon Major George Perry of the Indian Medical Service. Ludlow Perry studied medicine at St. Thomas Hospital, after which he joined the Indian Army Medical Service in 1895 and served in the Northwest frontier. After studying for a doctorate of public health DPH at Cambridge in 1906, he was appointed as the chief malaria officer in Punjab, also a lecturer in hygiene at Lahore Medical College, and as an acting sanitary commissioner to the Punjab government. Next slide, please. Now, it's important to place Perry's work in the larger context at that point in time, which is of this explosion in germ theory. Perry, as a malariologist, in part was following in the footsteps of someone in the same service in which he was functional, which is the Indian Medical Service, who is Ronald Ross, who had already won the Nobel Prize in the previous decade in the year 1902 for demonstrating the life cycle of the malarial parasites in mosquito. Germ theory was proposed and worked upon by Louis Pasteur in the year 1860s. The bubonic plague had hit Bombay in 1896. Cholera was a constant fear, and Robert Koch had discovered the cholera bacillus first in Egypt and then in Calcutta in the years 1883 to 1884. And the specter of the great influenza was just around the corner. But malaria was important because, unlike these preoccupation with bacteria, malaria deals with a macroparasite whose intermediate host, also serving as a vector, is Anopheles. And there seems to be an explosion of radiation of Anopheles in Jaipur. Next slide, please. And Jaipur, as Perry found out, is at 2,000 feet. And so altitude becomes important. And one of the questions he asked, and which continues to be of interest, is why might there be such an explosion and therefore greater propensity to the disease at 2,000 feet as opposed to the plains or higher altitudes of, say, 3,000 feet, which you can see in the map, like in Koraput, which is the district under which Jaipur falls. Next slide, John. And this is at the heart of the questions which Perry was asking, which stemmed from his observations pertinent to Jaipur, which were that, and I quote, "It is no exaggeration to say that to those who have knowledge or experience of the Jaipur hills, to think of those hills is to think of malaria." He's synonymizing Jaipur hills with malaria. Or that in passing from the plains of Visakhapatnam into the Jaipur hills, we pass abruptly from a region practically free from malaria into one that is notorious as the most malarious in southern India. Next slide, please. The observations pertain to the altitude of Jaipur and also with the resultant Anopheline diversity, some of which are shown here as representative of the 15 species that Perry recorded. These were found at different altitudes, which in turn influenced their feeding grounds and their feeding habits. So the two Anophelines to the left, Anopheles Jaipurensis and Maculipalpus, are stream feeding, whereas the other two, which characterize surface water feeding, which are found in the stagnant water, are Anopheles culicifacis and Anopheles. John, would you take forward?
Thank you, Ritija. Can you hear me? Cool. All right. So this is a slide. This is something of a divergence from citizen science, and we're going back 100 years, as you can see, a little bit over that. But what we're doing is pointing to an element of biodiversity that isn't really considered, as Ruth just said. And so if we could have we're having something of an example over here, and I'll speak more about this monograph a little later. If we can have the next slide, please. From I can do that. Okay. All right. So this is the beginning of Perry's paper, right? So you've got endemic malaria in the Jaipur Hill tracts of the Madras Presidency, and you can see how it is divided. And one key element over here is the distinctions. We're moving up from Vishakhapatnam, and you're going up about 40 to 50 kilometers, then you have this sudden rise, and you're going up to two plateaus, one which is 3,000 feet and another around closer to the Orissa border, it's 1,000 rising to 2,000. And you've got this, the major considerations over here, one's fed by perennial streams, the other ones are dependent upon monsoonal rainfall. So what Ruth just said is important because you have to think about those species that are stream dwelling versus the others that are surface. And so what kind of implications does that have as far as malaria is concerned? And so one thing is so one of the key conventional points of wisdom is that malaria is going to be found in standing areas, standing water areas. This is where we are, right? So is that what's happening? So what was interesting was they looked at what is known as the spleen rate. Do you have an enlarged spleen? And what I've done over here is actually say the spleen rate is a tool to classify malarial transmission and endemicity. And the spleen rate is defined as a proportion of persons of a defined age range in a given population that have an enlarged spleen and is expressed as a percentage. And what they found was that for the most part, as children, they didn't seem to, and even across sexes, there wasn't too much of a difference until you hit around 16. And then there were major differences across the board. And the way, and let's go to the third element. For recording the degree of the enlargement of spleen, 5 classes have been made use of. One from the costal central area, just a finger's breadth forms class 1, and then increasingly from that, the umbilicus is how you divide this into class 2, 3, 4, 5 in terms of determining what the spleen is, the spleen rate is. The spleen enlargement is. The spleen rates in an Aboriginal community, the term that he uses, Perry, there is a parasite rate of 80%. And this is the key thing. There seems to be no effect on the physique of children, right? So let's consider this for a moment. So this is, for example, the Garba men of this particular area and the women. And this is from that particular paper again. And here's what's interesting. There are immigrant groups to the area. There are those that are deemed Aboriginal, having lived in a particular area for a while. You find the spleen rate to be higher in those that are Aboriginal, yet the carriage of the disease seems not to affect people over there. Now, it's immediately interesting to think about what this means from the perspective of natural selection, right? And think about, is there a level of adaptation? Immediately ideas of, notions of, say, sickle cell anemia come to mind, and why is it that you will deal with this with malaria and so that, or Tay-Sachs, for example, with Ashkenazi Jews. But remember when Perry is doing this. So here are the observations way before what we might think of as common knowledge is now available to us. But here's what he writes. If a European were to go, right, and live in one of these villages amongst these people without medical treatment and without protection from mosquitoes, his health would rapidly be shattered and he could scarcely hope to be alive at the end of 2 years. If we bear this in mind, we shall realize at once the reality of immunity in malaria. The Aborigine harbors large numbers of the malaria parasite in his blood when in childhood, but is in a high degree tolerant of its presence and immune to its effect. And I draw this to your attention. I would again think about the work of the Pulitzer Prize winning author Jared Diamond, and when he thinks about geographic range and what happens again from the point of view of who
actually gets affected and who doesn't. Now, one major element, both in his work and also in that of Dane Kennedy's in the Magic Mountains, and I will come to that very shortly, is this. There's a suggestion that above a certain altitude, you are free of a number of diseases. This was quickly debunked, but nonetheless, there's still the sense that sanitaria are located up in the hills. So a number of the Pasteur institutes, for example, were up in the hills. You can think about the fact that TB sanitaria are located over there. Cholera was supposed to be escaped from if you got it later. Then there were major outbreaks in Simla and the like. Malaria, above a particular threshold, was not supposed to be seen. But what happens in these areas, which are these in-betweeners? 2,000 feet, 3,000 feet. Are you outside that range? Think about the northern parts of the country, the Terai, when you're going through forests, again, deemed to be largely malarious. And so here's somebody who's come in this, who's worked in the Punjab, has come down and makes these kinds of comparisons, right? So, yeah. So here's what Kennedy's saying. Not able to proclaim the highlands fully free, some were in the Anopheles mosquito zone, where it was possible to contract an especially virulent strain of Flaviatilis. Of course, those infected manifested no symptoms. So those were immigrants versus those that had already a certain level of natural selection playing in. What are our significant takeaway points? One, altitude alone is not a significant guaranteed escape from the causative agents of epidemic disease, particularly when weighed against temperature and rainfall patterns, as well as water retention and runoff on the ground. It's not just the rain, it's what happens thereafter. Second, adaptation to parasite load, however, is a feature. Human groups with long-standing associations show greater resistance to disease than more immigrant groups. So those are our two major points to take away. Now, I'm going to come back to this for a second and look at this. A monograph of the Anopheles mosquitoes of India by S.P. James and W. Glenn Liston. By the way, two of the Anopheles are actually named for these people. There is an Anopheles Jamesii and an Anopheles Listoni. And they're writing this in 1911. 1909, you have Thurston and Rangachari writing The Castes and Tribes of Southern India. I'd like you to think with us to locate the studies against these works that have emerged at that particular point in time, as you're talking about Aboriginal groups and the like, and also this massive diversity, this, as Rituja said, this explosion of radiation that has occurred. What is interesting, however, and this is part of the work that I've done earlier, is that when you come to the Fauna of British India series, while Liston and James are doing this work, the diptera dipteran studies are not including the Anophelinae. This will happen only 20 years later by Samuel Ricard's Christophers. And it's interesting to see that there is this disjuncture, particularly at this moment, as Rituja has pointed to, of germ theory making such a major splash at this particular point in time. Thank you very much. Questions?
Thank you, sir. If there are no pressing questions, we can break for a tea and questions can be asked during the tea time. That's okay.
Thank you, Rituja.
Thank you, Rituja.
Thank you, John. Thank you, everyone.
We have a 10-minute tea break. We'll get back in 10 minutes. Thank you, sir.
Check, hello. Check, hello.
You've done your molecular studies also? Which way, where you do that? Which way are you placed? Huh? Oh, good. Have we here then? Okay. आज ये कोशिश क्या है एक बार के दिन आपसे बात करेंगे मुझे आपकी एक सलाह चाहिए एक बहुत ही विचित्र मैटर के ऊपर सी आपको पता है मैं आजकल चेन्नई में काम करता हूँ नेशनल बैलेंस के अथॉरिटी के चेयरमैन हूँ नहीं पता तो मैंने डब्ल्यूआई से रिटायर होने के बाद अगले दिन सरकार ने मुझे ये सरकार की अथॉरिटी है नेशनल बैलेंस की अथॉरिटी और चेन्नई में स्थित है वहां पे मेरे को अपॉइंट कर दिया अब क्या है हमको ये एक हायर बिल्डिंग में हम लोग हैं अभी और अब हमारी तमिलनाडु सरकार से बात चल रही है कि वो हमको एक जमीन अलॉट करने वाले और हमारा 22 मई को अंतर्राष्ट्रीय बायोडायवर्सिटी डे होता है और हमारे केंद्रीय मंत्री वगैरह सब आएंगे तो अब मैं जो बदल चाह रहा हूँ ये है कि फॉर्मली तो सीपीडी सिस्टम को कहना और करना अभी काफी समय लग जाएगा बट मुझे क्या है कि वो लोग यहाँ का लोकल पहाड़ा है 30 सेंट बोलते हैं 1/3 ऑफ एन एकड़ 1/3 ऑफ एन एकड़ और वो अन्ना यूनिवर्सिटी है यहाँ पे चेन्नई के बीचों बीच उसमें वो जमीन हमको अलॉट करेंगे और हमको उसमें पत्थर भी लगवाना है तो फिर वो पत्थर तो मैं लगवा दूंगा मैं ये जानना चाह रहा था कि कोई रफ एंड रेडी क्विक कौन बता सकता है कि क्या एस्टिमेट होगा अगर 3 मंजिली बिल्डिंग बनाए तो 10 की बनेगी 20 की बनेगी 15 करोड़ की बनेगी कुछ आइडिया तो करना चाहिए उसका सो व्हाट इज दैट एजेंसी जिसको आप जानते हो हम उससे बात कर रहे हैं जो थोड़ा बहुत पैसा पड़ी लेंगे पर ब्रॉड इतना तो बता दें कि भाई 1/3 ऑफ एन एकड़ के ऊपर आप बिल्डिंग बना सकते हो 3 मंजिल बनाओगे तो ये होगा 4 बनाओगे तो ये होगा सम बॉल पार्क फिगर कुछ बता दो और कोई अगर चेन्नई में बेस्ड हो कोई आपका मित्र या कोई एजेंसी और भी अच्छा है उसको दिखा भी सकते हैं हाँ है ना प्लीज है ना हाँ तो वैसे तो सीपीडब्ल्यूडी करना तो आप भी प्राइवेट टीम के साथ ही करते हो पर वो आपके साथ में आते हैं वो मैं बाद में देखूंगा कि उसको फिर सीपीडब्ल्यूडी का बदलना कैसे पहनाना है पर आप प्लीज मेरे को के जरा बताओ इट हैज टू हैपन वेरी फास्ट है ना 1/3 ऑफ एन एकड़ इन अ प्राइम लैंड इनसाइड अन्ना यूनिवर्सिटी जस्ट यू नो कोई चेन्नई बेस ताकि हम कह सकें कि हम जाके मैं साथ चला जाऊंगा लिखा दूंगा तो ये है अब जरा सा आपने मैंने आपसे देखा था आपने कुछ स्पेस लिख के हमको थोड़ा बहुत ऑप्शन देना या कोई हो जाए तो मैं विल वेट फॉर योर थिंग। हां ठीक है मेरी मदद करनी है भैया। थैंक यू।
Hello, everybody. Hello, everybody. We'll continue our section. Our next speaker is Dr. Pravin Kumar. He's a postdoctoral fellow in IISER Tirupati. I invite Dr. Pravin for the speak.
So today I'm going to talk about part of our research project, ongoing project. So the title of my talk is Rediscovery of the Critically Endangered Alveolar Rat in the Eastern Ghats of Tamil Nadu based on geometric and morphometric genetic analysis. So generally rodents are largest group of mammals and it's widespread in nature and it's across all landscapes. When you look at India, it is 2,281 species that are there. And many of them are, considered suppressed. But when you look at the extinction history, in the last 500 years, 52% of the rodents are extinct, especially in Asia and Africa. So in India, we have 107 species of rodents, including rats, and 56 are endemic to Western Ghats, and 6 are endemic to 56 are endemic to India, and the 6 species of rodents that are endemic to Western Ghats. So when we look at these rodents, they are having a very wide distribution, but still there are few species that need conservation attention. For example, this kind of rock rats. So rock rats are living mostly in rocky areas in parts of India, and also there are more rock rats are there in Australia and Africa. So when we look at this, in the Indian type of a rock rat, they have 2 species. One is the Kutch rock rat, and another is alveolar rat. The Kutch rock rat, their distribution are their distribution in part of Kutch and part of Maharashtra, Karnataka, and rocky areas in the part of Tamil Nadu. So they are having different habitat associates with the rocks. When you look at this kind of large rock rat or the alveolar rock rat, they are critically endangered species. They are known from a single location in the world. And as of now, we don't have any information about this kind of rock rats. Why? Because very less attention of these small mammals in general on rodents, and also these animals are living in some ecological niche adapted landscapes such as these rocks. So these are alveolar rats are discovered in 1929 by Ellerman, and after that there's no pictorial evidence, there's no photographs, and we don't have any details of these animals in our museum. Or whatever people, Britishers have researched on those animals, they're collected and kept in this British Museum in NHM. So when we couldn't access some of the specimen, but still, there's no proper information on such species such as these rock rats. So our idea is to understand whether this animal is still living in Tamil Nadu, is exactly the type locality, or the animal went extinct. So in this regard, so this is the distribution of these 2 species. The yellow marked ones are the common, the Kutch rock rats. The one on the red one dotted in the Tamil Nadu part of Salem is a rock, it's alveolar rock rat or the large rock rat. Why? Because larger body size, the tail is very long, the ear is long. And our main objective is to see whether the animal is still existing in the type locality or not. The second objective is whether we can able to access the, because there's a small confusion during this, early taxonomic time. They're told that the 2 species of rock rats living in same location, in this same location in type locality where we are studying of this particular area in Salem. And whether we want to see whether the study location have both the species of rock rats. If so, why they are different each other, and how we can able to genetically differentiate. The third is there any phylogenetic distinctness between the two species? Then fourth is to understand the anthropogenic threats and also try to initiate the conservation at local level. So this is a study area. So the study area is 52 square kilometer area in Sherora Hills in Salem district. So which includes 16, in south part of the hills have 16 reserve forest, in which one is called the Kurumbapatti Reserve Forest, which is in the south, in the south part of the hills. And Kurumbapatti, we have a zoo also. Kurumbapatti Zoological Park is there. So actually this entire area is shrub dominated, arid, mixed deciduous forest, and mostly dominated by small rocky sites actually. Wherever we go and doing the field work in those areas, mainly we are using the Sherman trapping to collect and collect the rodent to understand its morphology. So actually these are rock rats living mostly in rocks, but we also try to set traps in other areas because we don't have any picture of these animals, we don't have any background information. So we are trying to study this animal through setting up traps in different areas like agricultural areas and human dominated areas, and some of the forest patches, some of the rocky areas, some of the stone walls within the zoo areas. In whatever areas we have started putting traps in different areas with systematically. Other than that, we are also using some random trapping sites that we chosen in different areas in high altitude areas. In 2014 and 13 to 14, we started studying this about the animal, and we couldn't find any individuals due to lack of field work and low trapping effort. But in 2020, 2021, we started our field surveys, and still our field surveys are ongoing. So the traps were baited with peanut butter and placed in different areas. So generally we are keeping the traps in the evening, and we are checking in the morning. And morphological measurements of the animal, such as head body, the weight of the animal, head length, the tail length, all the general characters are marked, and the GPS of this location were marked. So other than we are collecting the morphological data, we are also collecting some of the genetic data, like some of the tissue samples, some of the fecal materials we are collecting from whatever data from all the rodents we are collecting. And air samples also we have collected. Other than that, we are also collected whatever the animals were predicted as alveolar rats. We also collected few individuals for specimen purpose. And we also used genetic markers to understand its genetic distinctness. So we used molecular tools. So some of the areas such as big kind of rocky areas, and some of the medium elevation forest areas, and some of the semi-dry areas, and some of the cliffs where we also used setting up our traps in those areas. And some of the hilltops and rocks in the high elevation of Serurai mountain, we setting up traps. And some of the low elevation areas which are just adjacent to human dominated areas, also we kept some traps. And in the Ghat road and nearby some of the mining sites, whatever possible way we can keep the traps, we try our best to keep our traps maximum. So we got some interesting results that actually we started doing field work, and we have started collecting data from people also. So far we have interacted with 50 village peoples and with 432 individuals about this animal, whether this animal exists or not. Also we talked with about understanding their local ecological knowledge. Also we used actually 242 Sherman traps, live Sherman traps we used in the entire study period. We got 156 small mammals and 26 different rocky sites, or unique rocky sites we place the traps to study the animal. So other than that, we also characterize the rock, what kind of rock they are, and what diameter. So many details we collected from that. And we also noticed the roadkill data from the the road that are passing from the low elevation to high elevation. So initially, this is a Kutch common, the Kutch rat. This is very common. So initially we got this rat from our field site. Then after, our continuous field site, we are studying to, trying to understand. Then we got the, alveolar rat. So the alveolar rat is very unique, and they are having the larger body size, and they are, eyes are so big, larger than, the other rats, and the tail is very elongated. So we compared this one with the type specimen from NHM based on the geometric, morphometric, and also the genetic study, and we came to conclusion that these are the alveolar rats that will still exist in our rocky areas. So this is our some of the major results saying that some of our the 5 individuals we got actually, in which these individuals are living mostly in the bigger, larger, some kind of this kind of boulders. They're hiding inside. They're nocturnal species. They're living mostly in the high altitude, less human, less disturbed areas, whether human or cattle cannot enter. Such kind of habitats only they are preferring. And other thing is they are mostly looks like they are mostly preferring some kind of a stone that's called charnockate stones. They are very chill in nature, so they're adapting to live in this kind of rocks. And these animals cannot dig a burrow, so only they can able to hide in between the cliffs, and they're adapted to live in this kind of condition only. And so far we collected, we have recorded the species from some 7 sites in the hills, and mostly female individuals we have noticed from the medium elevation to high elevation areas. And they are preferring large boulders, like like the 42 rocky areas we get found, in which all the individuals are mostly from large boulders only. And although we kept traps in stones, boulders, and some of the caves, but they are large boulders with multiple openings are the only preferring habitat for this kind of Elvira rats. So there are some threats we observed, and mostly grazing, human movement, and pollution are one of the threats. And we also had one interesting observation is when the elevation, like 689 is our medium elevation, that we had noticed the tail is not so much long, like a little bit same species only, a little bit short. When we go on the high elevation, the tail length getting a little bit higher. And some of the morphological measurements of this, it's close related species. And other than that, we also noticed some other, some of the species like white-tailed wood rat, Rattus rufescens, Criminum caticus, and musk species, some of the sunkus that are shrews, and the Criminum caticus shiva, it's a sister species of the Elvira rat. And our morphological measurements clearly, skull measurements is clearly showing that this, skulls are very unique, and they are very different from these close related sister species. Why? Because this bullae is small in size, and this palantal foramina is a unique factor. Palantal foramina, there's a gap between the teeth, so that one is extended between the tooth row. So this is one of the important character to differentiate these rock rats from all other species. So this is its close related species skull. So as they are, these both are Crimnomus, that means the rock rats. They both having unique character of this elongated palantal foramina. And our geometrical morphometric analysis, clearly, revealing that the species, the Elvira rat, having very, good or very elongated tail, and they are having a very unique differentiating character from its close related sister species, Criminum caticus. So why? Because when you look at these results, the head body length is very longer than its close related species, as well field as well as the museum specimens comparison. When we look at the tail is again very longer. So when we look at the brain case, actually, it is just 4 centimeter. Actually, look at the entire, entire animal length. It's almost, 123, centimeter entire head body length. Look at the ear length. Ear length is again, 4.5 centimeter. Larger than any, Criminum caticus. And when you look at this hind foot length, it is again very, prominent and very larger in size than its comparatively sister species. So we also compared the, the rock rat specimens based on their, their morphological measurements again. So what we compared is, this one is we compared based on our collections in the brain case. So this brain case is clearly showing that the species having very, unique way of, adaptation skills. And so this particular issue is saying that this PC is clearly showing that the field and field collections and the earlier museum collections are clearly showing a uniqueness with its it's very far away from the Criminum caticus details. And also this high bootstrap value is clarified from Criminum caticus from our field site, Criminum elvira from our field sites is clearly showing that there's a clear difference and differentiation and species distinct from each other. And this is some of a glimpse of fieldwork, but there are a lot to do more. Like we have to understand the usage of pesticides in and around those. Actually we have 67 hamlets in on that particular site, and we need to study more about rodents usage in those areas, and we had to understand its population, how many individuals of failure rats are there. So now we are planning to start the mark recapture method to understand its population in each grade. And we are trying to understand the conservation priority sites. So we want to map the conservation priority sites and understand illegal logging, because these are living in high elevation mountains and adapted living by a lot of bamboos and other high dominated native trees. So we need to understand how much timber logging is affecting the species directly or indirectly, and monitoring the habitat and population estimation is high priority. And these are animals are still listed as a vermin in the Indian Reproduction Act. So it is should be considered as separate important species in protected categories. And there's a lot of policy intervention is needed as of now. So establishing village forest committee, we are just started with our capacity building program with the district forest officers, conservation officer. Also we are giving training to range officers how to understand differentiate these species from other species. Like we are conducting regularly meeting with villages also. So policy level intervention. Other than that, we have to include these animal and these details in the Salem district level working plan. And also there's a lot need to be done about the roadkill management, because we are thinking that a lot of every month we observe that 20 to 30 individuals of roadkill happening, especially small mammals in this ERCOT high elevation area. So also we have to understand more about its road and vector borne diseases, and the inclusion of this Elvira as a priority species for all the conservation efforts. And major financial support from Cetacel Edge and Idea Wild, and for the camera, for the equipment support, and part of the work supported by this DST SEB and PDF project. And 2014, I got WF India Small Grant, and they are the initiative fund supported through my research. And I like to acknowledge Dr. Sanjay Muller for his help, and all lab members, BNHS, NHM, and forest officials of Tamil Nadu for supporting and helping in this carry out this research. Thank you. Any questions?
Thank you, Pravin. We can maybe take one or two questions. We're running out of time.
Very interesting study, I would say. First time I'm hearing about the species over here. But where have you reported it?
It is from the Kudumbapatti Reserve Forest.
No. In scientific literature, have you already reported it?
We are just submitting, sir. We are just submitting.
Okay.
Yeah.
Okay. And then you have plans for obviously carrying on with the study?
Yes. We are planning to continue the study.
So, I mean, what appears to me, maybe you are doing it, but microclimatic variations, maybe they can provide some clue.
Yes, sir.
Because see, we all measure temperature, rainfall, this, that, whatever, but there might be some factors operating at that level, which need to be very intensely studied.
Yes, microclimatic.
Yeah, and what about complete genetic profiling and things?
Actually, we are just a part of the sequence only. We just part of the gene only we tried, but we are planning to use more, genetic markers to test its nuclear genetic types also. But that is also coming soon, sir.
And you're sure outside Tamil Nadu or the Salem, they are not reported anywhere?
They are not reported. Recently, we had trip to Gujarat and Rajasthan. We carried out extensive surveys in some areas in Gujarat, Rajasthan, especially in the Kutch and other areas. And we couldn't find this kind of particular species like these Elvira rats. But there are Kutch rats are there. We sampled some individuals, and that is still in molecular analysis. So maybe more.
And who is the PI of your project?
Dr. Nandini Rajamani.
No, that's fine. See, I am more keen of its implication and reporting and recognition and so on and so forth. And obviously in our country, ZSI is the agency which also has to be informed that such and such discovery, recovery has been made. So those processes also need to be initiated fairly early. And of course, his intellectual property rights on that kind of a study is also, very important. It is there. See, this first talk is going worldwide. So I am very concerned about his ownership of this kind of a thing. Yeah. Okay, good work. Please do that.
Yeah, thank you. Thank you.
If there are no more questions, so the next talk is by Mr. Rajashekar Banti, the citizen science coordinator at IISER Tirupati. Yeah, sorry for that. And the next talk will be by Ms. Chitti Aravind, PhD student at IISER Tirupati.
Hello, everyone. Is my screen visible? All right. Hi, everybody. My name is Chitti Aravind, and I'm a PhD student with Dr. Robin. And today I'll be talking to you about how we created a detection framework to detect the critically endangered Jordan scoser. So using bioacoustic technology. Now, why really bioacoustics? Monitoring techniques are essential in the conservation of species, and in the case of birds, point count surveys were the most popular method. However, with the advent of upcoming technology, bioacoustics is proving to be an excellent tool in biodiversity monitoring. And there have also been several comparative studies to show that using recorders make FAIR as a better tool. A few advantages of using recorders are that we can cover a large area. They are also programmable. The data can be stored and retrieved later for analysis, and there is also very less human interference in data collection. Several studies have been used using bioacoustic technology for the monitoring of birds, and these include nocturnal birds, birds with infrequent calls, and also endangered birds. Our lab has previously extensively worked on bioacoustics, and we've thought this would be a great opportunity to use bioacoustics for the monitoring of the critically endangered Jordan scoser. A few facts about the Jordan scoser first. It is a cryptic, nocturnal, and elusive species with also an infrequent calling pattern. It is listed as critically endangered under the IUCN Red List and is also a Schedule 1 species. It is endemic to a tiny pocket of open scrubland habitat, which is found in the foothills of the eastern Ghats in Sri Lankamaleshwara Wildlife Sanctuary, which is in Kadapa in Andhra Pradesh. The last confirmed sighting of this bird was decades ago, way back in 2008 by Dr. P. Jagannathan, who has worked extensively on this species. Let's jump into a few methods that were previously used for the detection of the Jordan scoser. The most popular method was by using camera traps, and the other method was by using tracking strips, which basically are strips of sand laid out on the soil, and footprints were used to match the footprints of the Jordan scoser. These techniques were useful in the detection of the bird, but might not be efficient long-term monitoring methods. And so we decided to come up with a detection framework using acoustic technology. Using acoustic technology usually involves two main components. One is the hardware, which are our recorders that are used for recording the soundscape. And the second component to this is using software, which is basically a means to analyze the collected recordings. This brings us to our objectives. The first objective was to create a robust field protocol for the installation of automated recorders. And this involved two main components. The first component is to conduct an attenuation experiment to infer how far apart recorders are to be placed from each other. And the second component was to actually program the recorders and deploy them at our study site. The second objective is to create a detection framework, as I explained before, using commercially available software. And this also involves two components. The first is to create a recognizer for the Jordan scoser's call, and then to use this recognizer to screen the collected recordings. So for the first objective, here are the four types of recorders that we used. The first is a song meter 4. The second is a Swift recorder. The third is a rugged Swift, which is essentially a Swift in a hard casing that can also house more batteries. And the fourth is an audio Moth. So for our attenuation experiment, so why did we need to do this? This is because sound attenuates in different habitats based on the vegetation present, and is important to account for sound transmission properties to discern the spacing between recorders in any habitat. So what did we do for this? We actually placed all our recorders about 1.5 meters above the ground in a particular location, and then conducted playbacks of the Jordan causer's call at various distances from the recorder to see till what distance was the recorder able to capture the noise of the bird. And what we found was that all the recorders recorded till about 500 meters and 2 of them recorded till 700 meters. So we conservatively decided that our recording radius would be 500 meters, and in turn, our grid size layout was decided to be a 1x1 square kilometer grid. Now, for the second part was the actual field deployment. Here you can see a map of Sri Lanka-Malleshwara Wildlife Sanctuary enlarged in the larger map, and we gridded up the core area with 1x1 square kilometer grids. Around 17 recorders were deployed at a time. All the dots represent recorded deployment locations, and the yellow dots represent locations where the Jordan's causer has been previously sighted. Our recording programming schedule was scheduled to record from dusk to dawn, a nocturnal schedule of a continuous 13 hours. A sampling rate was set to 44,000 hertz, and the duration that we conducted our study was for a period of 5 months from October 2019 to March 2020. We also conducted monthly visits for battery changes for the recorders and also SD card changes to retrieve the data. So now once we have got all our data, what we need to do is analyze it. And for this, we needed to create a call detection framework using commercially available software. But before we do this, we need to know what are the species that are present in the Jordan's causer's habitat. So to find out all the co-occurring species, we obtained a list of all the birds present in Sri Lanka-Malleshwara Wildlife Sanctuary and shortlisted all the vocalizations of birds that are nocturnal and call within the Jordan's causer's frequency bandwidth. Once we had our list of birds and their vocalizations, we proceeded towards creating our recognizer. So for this, we used 2 different software. One is Raven Pro, that is developed by the Cornell Lab of Ornithology, and the other is Kaleidoscope Pro, developed by Wildlife Acoustics. The Raven works on the principle of spectrogram cross-correlation, and Kaleidoscope Pro works on the principle of k-means clustering. So to create a recognizer, especially in Raven Pro, we need to select a good template of the Jordan's causer's call. And for this, you can see here is a spectrogram representation of the Jordan's causer's call. And it is essentially a disyllabic call, which is that the particular song has 2 notes. For our purpose, we used each note as a specific template. So now when we use this template, we use this template on a test data set and also see the performance of our recognizer. Now to evaluate the performance of our recognizer, we use a number of different performance metrics, most importantly, precision recall, and also an F-beta score, which is a harmonic mean of the precision and recall. Now, in the case of an endangered bird with an infrequent calling pattern, we do want to focus on precision, on recall rather than precision, and we set the beta value to fall as 2. Once we have evaluated our performance of the recognizer, we actually use these 2 softwares and the recognizers created in both software to screen the entire data collected. And once the data is screened, we receive a number of detections from both the softwares. Now, each of these detections need to be manually cross-checked to categorize them into calls that we know, which might also be the Jordan's causer. And in certain instances, we did receive calls that we were unable to classify. So we created a questionnaire of all these calls and sent them to 11 experts all over India who are famous, who are very well versed with the bird species of this area, and asked them to rate the call of whether it would be most likely to belong to the Jordan's causer or least likely to be a call of the Jordan's causer. And then we also furthermore performed a spectral cross-correlation of these unknown calls with the Jordan's causer's call. So moving on to the results of our field deployment, We collected about 8 TB of data, which is about 24,000 hours of data. And we evaluated the performance of all the 4 different hardware recorders that we used. And we found that the Swift rugged recorder worked the best for us, also primarily because it has the capacity to store 12 batteries and runs for the longest duration of time. So at a particular stretch, it can cover around, it can collect around 700 hours of acoustic soundscape. For the results of our recognizers, we found that the Raven Pro software had a much higher precision recall and FPTA score. So this worked better as compared to Kaleidoscope Pro for us. And we had a number of species that were detected by our template detector that were co-occurring species in the Jordan's Scosus habitat. So a few of them are the Jordan's nightjar, the Savannah nightjar, and the Indian nightjar, the common hawk cuckoo, the Indian peafowl, the gray jungle fowl, and also the shikra. But we also received, as I mentioned earlier, some unknown calls. And here is an example of an unknown call that we received, which looks very similar to the Jordan's Scosus call. And what we did in this case was that this was a one-off instance. So to actually see what this call was, we examined the context of the call by checking out the recording 60 minutes before and after the call. And what we saw was something really interesting. We saw that there were calls of the bayback shrike in several intervals, and these were interspersed with calls of the Indian thickney, the Jordan's Scosus-like call that is shown above, and also the bulbul. And we know that the bayback shrike is a very good mimic. So this made us believe that this Jordan's Scosus-like call could be the potential mimicry by the bayback shrike. Just having this one instance does not enable us to draw any conclusions, but it does highlight the importance of examining mimicry also as a potential source of identifying such endangered species. So moving on to the outcomes, we were able to create a robust tested framework to detect the Jordan's Scosus using automated recorders. Our recommended field protocol is to stick to the 1x1 square kilometer grids and use rugged recorders. And we also do recommend using Raven Pro as software to create a recognizer for this particular species. A few limitations were that there can be a high investment of the recorders itself. Recorders can also malfunction, leading to loss of information. And also there might be variability in attenuation properties due to differences in vegetation patches across the landscape. Future directions are that our team of researchers from our lab are also planning to expand this research and examine 3 reserve forests, which is Yerpadu, Golapalli, and Venkatagiri, and also use automated recorders for the detection of the species. I would like to thank the funding sources, the Wildlife Conservation Trust, ISA Tirupati, and the Nature Conservation Foundation, the Andhra Pradesh Forest Department for their support, Mike Prince for the audio mods, the EcoEvo labs at ISA Tirupati and at Cornell University for feedback. And of course, nothing could be done without our project volunteers who helped us for all the field work during the field sessions. Thank you, and happy to take questions.
No, it's not a question, it's a wonderful initiative. And thanks for taking so much of interest in initiative. From the forest department, we have invested a lot of our reputation, time and money in trying to look at this elusive bird without much success. And you have given some hope, and please continue the work. And the other alternative areas which have selected, the Venkatagiri and other places, continue the work. All the best to you.
Thank you.
Are there any other questions? I guess not. Thanks, Chitti.
Okay. Yeah, thanks.
So the next talk will be by...
One quick question. When you said high cost of that equipment, what is high?
Chitti?
$10, huh? I mean, high, but not... Yeah.
Should we move on? So the next talk will be by Ms. Archita Sharma, PhD student at ISA Tirupati. Is the slide visible as slideshow? Okay, all right. So I'll start. So good morning, everyone. I'm Archita. I'm a PhD student at IISc Tirupati. I joined last year. And today I'll be talking about the undiscovered potential of examining cryptic avian diversity in the Eastern Ghats through genetics. Now we know that species are the standard currency of biodiversity assessments and conservation planning. But what are cryptic species? Cryptic species are two or more distinct species which are overlooked due to morphological similarities. And so they are often classified as single nominal species. To put simply, cryptic species are hidden species, and they are measures of undetected diversity in a landscape. Surprisingly, cryptic species are distributed across taxonomic groups, and one of the best examples comes from India, which is on Western Ghats laughing thrush and Sholicola. So both Western Ghats laughing thrush and the Western Ghats shortwing remained a taxonomic confusion for long. So on the left side, you can see a map from Ali and Ripley in 1996, where they have put Kerala laughing thrush as two species of Garrulax, and one of them has 3 subspecies. But later on, Robin et al. Published a paper in 2017, which revealed with genetic data and also data from plumage and songs that what we actually have in both these groups are multiple distinct species, which diverged quite early, almost 5 million years ago. And this was the first evidence of in situ avian radiation from the Indian subcontinent and revealed endemic species in the Western Ghats. Now let's come to Eastern Ghats. Another notable study by Ramachandran et al. 2017 looked at the peninsular Indian subspecies. They wanted to look at the distribution of different races and subspecies across peninsular India. And they found 112 species, including 20 monotypic ones, and 117 subspecies of birds from Eastern Ghats. These numbers do not include waders and migrants. And what's also interesting with respect to biogeography of Eastern Ghats is the close proximity with both Himalayas and Western Ghats. During the Oligocene, most of the peninsular India was forested, but following Oligocene, much of the path started drying. And the Eastern Ghats might have served as an important moist forest refuge for both Eastern Himalayan birds and Western Ghat birds during these glacial cycles. And one of the examples we can see is in the contemporary distribution of yellow-browed bulbul. We can see an isolated population of yellow-browed bulbul in the Eastern Ghats. 2 more examples of geographically isolated populations in the Eastern Ghats are these 2 babbler species, puff-chested babbler and Abbott's babbler. Both of these have affinities with Northeast India and Southeast Asia, but you can see how they remain and have continued to survive in a small pocket in Eastern Ghats. Interestingly, Eastern Ghats also has major biogeographic divides, the 3 of them being Godavari River, Kaveri River, and Mahanadi River. The Godavari River is one of the major biogeographic divides across peninsular India. It serves as both a physical and climatic barrier. A 2008 study found that this barrier is almost the Godavari basin is 130 kilometer wide. And it also marks as the southern boundary of moist deciduous forest in the Eastern Ghats. But to really look at the impacts of these barriers, let's move on to the subspecies which are distributed across Eastern Ghats. So we have 23 species with more than 1 subspecies across Eastern Ghats. And here I want to bring out the example of tawny-bellied babbler. So it has 2 subspecies in the Indian subcontinent, and you can actually see from the photo that they are morphologically distinct. The one above Godavari in Odisha lacks a white throat. But in the photo that's down, which is the subspecies Albogilaris, has a distinct white throat, and it also has gray eyes. The subspecies above, Hyperethra, has darker eyes. Another example would be golden-fronted leafbird. You can see that here the biogeographic divide is Mahanadi River. You can see populations which are in Odisha, in fact, also in Northeast and extending up to Southeast Asia, Thailand, have this golden rim around the black throat. But this is missing in populations which are south of Mahanadi and this subspecies Insularis. Another example where the river Kaveri acts as a biogeographic divide is common Iora. Here, the difference lies in the male bird's plumage. The birds which are distributed in the southern tip of India and Sri Lanka are constitute a subspecies called multicolored. They are more black and they only have one white band on the wings, whereas the subspecies which is found north of Kaveri have two white bands, and they're also slightly lighter when we look at the proportion of black on the back and below the head. But how do we test the intra-species variation, or what's the genetic distance between these subspecies? The only way is to use molecular data, which can actually help us quantify the impacts of these biogeographical barriers. Even comparatively well-studied birds, well-studied groups like birds can yield well-differentiated and evolutionary distinct lineages. And these are often found in species which have widespread distribution. Several studies using genetic tools globally have discovered cryptic diversity and species level differentiation in areas with unexpectedly no number of endemic species. The research workflow in our lab employs systematic geographic sampling across the distributional range of a species. We deploy mist nets. The capture of birds using mist nets is a standard protocol used to monitor avian populations worldwide. And we also have developed standard protocols for DNA extraction and taking morphometrics. Then we plug this into lab work and sequencing, and finally phylogenetic and biogeographic analysis, including divergence time dating, and for cryptic lineages, integrative taxonomy, where you employ genetics, morphology, and behavior like calls to actually test species limits. Here, I'm just providing a preliminary genetic analysis of intraspecies diversity of Indian scimitar babbler. Over the past decade, much of the phylogenetics has relied on multi-gene markers or using mitochondrial and nuclear gene markers covering max to max 15 to 20 loci. But with next-gen sequencing, you can cover genome-wide several loci. So here we have used about 4,000 UC loci to look at the evolutionary relationships of individuals of Indian scimitar babbler across peninsular India. And we see that here in this tree, Goa gap does not seem to be a major barrier for Indian scimitar babbler. Also interesting is the sample from Nilgiris falling with samples north of Goa. These are disjunct geographic locations, but still we receive this clade with high support. But at the same time, I don't want to overinterpret this tree because we still require more individuals, and we are missing a key subspecies of Indian scimitar babbler from Eastern Ghats. And that would actually help us build a complete picture of how Indian scimitar babblers and their respective populations are actually related to each other, how they could have colonized different locations. So yeah, getting samples from Eastern Ghats remains a priority. And that's, so I'll just come to the future scope. We do want to do systematic field surveys and DNA sample collection to examine genetic diversity across multiple avian families like flycatchers, babblers, thrushes, pipits across Eastern Ghats. This would help us reconstruct evolutionary relationships within a global phylogeny using multi-gene and UC data. And it would provide first genetic description of several races and subspecies of birds across Eastern Ghats. One of the major limitations of biogeography project has been getting permits. We are waiting for permits to start field sampling in the Eastern Ghats. It would be great also to have multiple species across these different families so that we can build a picture, a community picture, or really understand evolutionary patterns at the family level for birds. The expected outcomes would be, we would get a robust estimation of alpha diversity when we uncover potential cryptic lineages with multi-line evidence. We would also be able to quantify role of different barriers in the Eastern Ghats. And also presently, conservation planning and management relies on accurate species level taxonomy. Subspecies do not get conservation plans, which is why it's important to understand what are the intra-species variation and to identify and uncover cryptic species. And that brings me to the end. I would like to thank members of Shola Sky Island, ISIP Tirupati, forest departments of Andhra Pradesh, Kerala, Tamil Nadu, Karnataka, Maharashtra for both past and ongoing field sampling, and funding support to Dr. V.V. Robin from SERV. And I would like to thank everyone for listening.
Are there any questions?
Sorry.
See, all these are fine, but today when we discuss, we cannot afford to not to talk of climate change. So every study must be in some ways could be contributing or potentially looking at how climatic variations might also affect this cryptic behavior that you are studying. Any thoughts on that?
Yes, I would like to point out even across Eastern Ghats, we have different climate regimes. The north and south are very different. For example, the moist and the colder climate in the northern Eastern Ghats supports a population of Malabar trogon, but the southern Eastern Ghats don't. So climate is important, and it would be interesting how even species track climate and could be changing their distributions as the climate changes. But yeah, for that, we would need to ring birds, ban birds, and just monitor bird populations for a long time.
Fine. Let's look at your limitation slide.
Okay.
Unfortunately, at this point, I don't see any representative of the forest department. But what is the issue? For 4 years is a long time.
Yeah, we were waiting for permits, and so 2 years ago we did get a chance to sample in Andhra Pradesh, but we could only provide a small set of species and not as many as should have gone.
So what, according to you, is the issue? An understanding, or what are you going to do?
No, getting the permits is the issue.
Yeah, I think we don't know what the issue is. It just is just getting delayed. So if there is a specific objection or this thing, we can address it. But I think that there is a lack of trust, and so we are basically not allowed to work even in our campus. So. Sure, I'll do that. Sure. Sure, thank you so much.
Just a remark. Thank you, Dr. Mathur. And, this is a matter of great concern to academicians and to growing institutions like ISA. And I do remember that there have been, I was just asking Robin, have you represented again and again, met them also, but it did happen to us in Pondicherry University also, but those stages were different. But with the time, things have changed a lot. Over just a few minutes sitting across the table, PCCF just gave it to us for the large scale study in the Eastern Ghats. So with the time, things have changed, but I think there is a representation of fair group with the support of NBA. I think this is very much a necessity for the young research scholars and the project people, which are all time bound. When they wait, they can't wait any longer, and they have on other side compulsion from the funding agency side, compulsion from academic framework of PhD administrative aspects. So all these shouldn't be a stress to the scholars, because we need original data, and true Indian ecology needs to be rewritten with decades of contributions from our young, energetic, enthusiastic scholars before they lose that. So I think please keep representing with the support of NBA and other concerned agencies. And this needs, I think, a representation. So yeah, we also suffered this way, but re-representations did help for our 30-hectare Anamalai plot. And it happened with the 8-hectare Koli Hills plot. Bharat managed it. I wouldn't tell here how.
Yeah, I think the consideration of the student's career is also very important. I mean, so the PhD student who's on this project, his PhD was getting delayed. So finally, he just decided to get married, and last week he was married.
So if there are no more comments, for the next talk, we have Dr. Bhupesh Gupta, founder and director of Universal Eco Foundation, Pondicherry.
Sir, we are not able to hear you.
So audible, sir?
Yeah, now it is audible.
So I guess we can start.
Sir, please start your presentation, sir.
Yeah, 1 minute, sir. So audible, sir?
Yes, it's audible now.
Yes, sir. Just a minute, sir. My laptop got stuck actually. Just a minute. Sorry for the delay. Yes, sir. Vanakkam, Vandanam, Namaste, Namaskar, and good afternoon. I am Dr. Bhupesh Gupta, founder and director of Universal Eco Foundation. Actually, first of all, I thought actually.
Sorry to interrupt. Sir, sorry to interrupt. We are not able to see your slides, sir. Can you please present? Just stop the screen sharing and reshare it once again, sir.
Now fine, sir?
No, yeah.
Is it visible, sir?
Yeah, visible.
Thank you, sir. Thank you so much. So, Vanakkam, Vandanam, Namaste, Namaskar, and good afternoon. I am Dr. Bhupesh Gupta, founder and director of Universal Eco Foundation. I would like to talk about the status of fauna from Seshatchalam Biosphere Reserve, Eastern Ghats, Andhra Pradesh. Actually, earlier I was working in Andhra Pradesh Forest Department as a biologist, but I don't have any separate funding agency. I don't have any separate funding. This is my own interest. So when we were writing the management plan, so we need actually support documents like what are the checklists, what are the species available in the Seshatchalam. So what are the threatened species is available in Seshatchalam. We don't have anything there. That's what it's my own interest. I started for the inventory studies. So based on the data only, now I'm going to present. So you know this Seshatchalam. Seshatchalam is the first biosphere reserve in Andhra Pradesh. It's located in Chittor and Kadapa district. You know very well about this. See here we having almost dry deciduous forest pages. Some of these places having for moist deciduous like Talakona area, Kankumadi area. Those areas you can see the moist deciduous patches also. But I am not working for the entire Seshatchalam biosphere reserve. I worked almost for 150 square kilometer area, especially for Chittor area and the border of Kadapa we worked actually. So based on that result only, actually I come across with the checklist and everything. So we can see the Seshatchalam biosphere reserve. We having almost 4 divisions, the main divisions, the Kadapa, Rajanpeta, Wildlife Management Circle, Tirupati, and Chittor East. This is a major zone for the red sandal as well as the biosphere reserve. So first I thought, what are the species or present? And the second one is, what's the status of the species? So this is the major important for the conservation and other aspect. So that the basic data is very, very required. That's what I started for the 2 options. The study duration, the mammals, I started for 2012, 13, 14, 15, and 2019. And Herpetofauna 2012, 13, 14, 15, and 2019. Birds 2011 to 2014, and butterflies 2012, 13, and 14. So you can see this totally entirely done for this direct count methods only, sir, actually. Whenever visiting the species through camera trap as well as my camera, we took that only. So I am also presenting that what are the photos is available. Those only I'm going to present. There are so many indirect evidence also there, but I am not considered in this presentation. First, we done the science survey, sir. Where are the indirect evidence is available? Those area we made it for the grid. Particularly this area we fixed for the different kind of camera trap. We fixed for the Gedipe camera, Moultrie camera, as well as pipe end cameras. So in that, a total of 26 species of mammals belonging to 17 families were recorded. The IUCN status like the Bengal tiger, this is the first report in the Spatial Bias reserve. It is taken in the near Talakona, almost 5 kilometers from the Talakona. We got in our camera trap. Also, though it's quite common here, Asiatic elephant almost we reported, but we don't know exact number. But 26 to 28 individual we recorded, but more than that also might be there. So Indian pangolin, sloth bear, sambar deer, leopard, rusty spotted cat, and grey slender lords. This is the threatened species have been reported during our studies, sir. Also, this is the camera trap photos we captured from the Wallabulli range, as well as for the Mamuntur range, and the surrounding area we captured so many elephant also. Five types of anglets have been reported: spotted deer, sambar, four-horn angler, mouse deer, and barking deer. These are the 5 deers variety also reported in this Spatial Bias study, sir. Also, bonnet macaque and grey lumpers, so wild pig, Indian hare also reported in our bias study. So this is our photography, my direct photography. So that's what already I mentioned in the direct my photography as well as camera. This only I considered this whole study, sir. The giant squirrels as well as slender lorries. So also leopard captured, there are many places, but we don't know exactly what is the estimation of leopard, but what are the species available in the Sashachalam, that purpose only we done. So also this is, I said already, this is the first capture in Sashachalam for the camera trap. So recently we got in 2019, almost the last year only we captured actually this Bengal tiger. Also rattle also captured, quite common here, and sloth bear and wild dogs also. So wild dogs, it's frequently you can see the healthy wild dog also in our Spatial Bias study. So also small Indian civet, calm civet, and rusty spotted cat and jungle cat also have been reported in this my camera trap study. And ruddy mongoose and grey mongoose, golden jackal, and porcupine also reported. So compared to the birds of Spatial Bias reserve, because of we don't know anything for sufficient data is available in Spatial. That's what we started for each and every taxa for proper methodology. So almost we followed for the systematic methodology because of we don't have any proper we don't have any line transect method or something like that. We have some curved line also. Wherever possible to walk, those areas to walk, and we encounter the birds have been listed out, and finally we analyze. So more than almost 215 species of birds and belonging to 45 families have been reported. The yellow-eyed warblers, so yellow-throated bulbul, greater spotted eagle, and dotted painted stork, Paleo hairy, Alexander parakeet. Some of the migratory birds timing you can see, especially for Karakambadi dam as well as Talakona inside, there is another remote dam also there. In Tirumal also, you can find especially for the daughter also. And the Karakambadi dam, you can mostly will encounter for the painted stork and Paleo harrier also. This is a near-threatened species also reported. Also, using sectors gathered, this all the photography captured during my inventory studies. Also coming to reptiles, because of reptiles also lack of our checklist. That's what I want to start the reptiles also. The reptiles also followed for the visual encounter survey methods. So have been followed throughout the, especially for the International National Park as well as the adjoining the protected area. So I went 500-meter length transect were covered on by foot, and each and every habitat also explored. So Herpetofauna 2012, 13, 14, 2015, and in 2019 also we continued for the Herpetofauna survey. So we know that method and how you are doing for the morphology of everything. So same methodology have been followed. A total of 83 species of Herpetofauna consisting 21 species of amphibians, 25 species of lizards, 33 species of snakes, and 4 species of Lelonian species have been observed during my study periods. So this is a Buffonidae species, 3 species, followed by Vitroidae 5 species, Raccophonidae, Diclorocinidae, and Ranidae species also have been reported. So have been published also recently the Journal of Animal Diversity. So also coming to reptiles, in the 25 species of lizards belong to 6 families were observed, and the Agamidae, Chameleonidae, Varanidae, Geckonidae, Cinidae, and Laceridae also have been reported in this research. We know very well about this. This golden gecko is almost 10 years back, Very rarely you can sighting. Now you can see the near Kabila Tirtham also you can find this golden gecko. Now quite common for everywhere for golden gecko. And this termite kill gecko and common garden lizard and the common sking is quite common everywhere. All the sites throughout the reported. So this is the lizard species have been reported our Statistical Bias studies. So also coming to the snakes part, almost 33 species of snakes belong to 6 family have been reported. So among these 6 species are venomous species. There are so many mild venomous also there. The Colibrid 21 species, Bovid 3 species, Elapid 3 species, 523 species, Typhoid 2 species, and the European 1 species have been reported in this biosphere. And some of the uncommon snake species also reported or Statistical biosphere, common sand boa, python. And most of the places, villages, there are unknowingly people are killing all these snake species. So we had interview with the local people. They said all these species are venomous. They thought still it's all these species are venomous. So the need to create more awareness is a very important for the local people. So understanding about this, which is the venomous, which is the non-venomous. So still the people are getting confused and they're killing so many species also. So this is a species have been photographed and published in the Statistical Bias. And this is one of the recently reported for 3 years back, reported for the Sri Lankan flying snake. This is the first report in Statistical Bias. And this is also recently published in sand snake. This is also first report in the South India. And this is brown wine snake. This is the first new locality records in Eastern Ghats. And some of the specimens have been compared with our biolab. This is Nagarjuna Sagar Srisalam. This is the first report in Nagarjuna Sagar Dam. The second is we reported after 35 years in Statistical Bias. I said already this is the 6 venomous species have been reported. And coming to the butterfly study, because of this also government needed to put in this management plan. That also we started for especially for the what are the species of butterflies available in this Statistical Bias. This is almost we started morning and evening. Almost we reported 96 species in Statistical Bias. This we are followed by the belt transect method. So almost a length of almost a 500 meter length and left and right side for 5 to 10 meter either side. So what are the species you encountering? Those species we were reported and photographed also in the morning and the evening. So this is some location we've been identified. So almost those places where are the species available. So those species were photographed as well as you would encounter also the number, but you could not calculate it for properly. But first of all, we need it for the what are the species available. That only you took in the list. So this is the family-wise you were recorded, and the Papillary 9 species, Peridae 19 species, the Ephylidae species, Vicaridae and the Hesperidae species also reported from the Statistical Bias. And the number of genus and species, common, very common, rare, and uncommon, based on the numbers. So we given this occurrences. So even habitat wise also you plan to do, but you are not continued for the habitat wise. But this is the lack of Statistical Bias. In future, somebody want to do or something like that, the habitat study is the very essential for the butterflies to improve our habitat. So this is the species have been captured in the Statistical Bias. So the main problem is the main threat is the ignorance of pilgrims who quick inside the forest. This is the one side the big problems we encounter for every year. The another thing is the red sandal smugglers who divert the forest department attention. So they're making fire for the those under here. The people are going forest department, people are going one place to other place. That same time they're cutting all the trees, red sandal and everything. This is the major problem we encounter. Some of the smugglers also inside there. So anybody want to do research or something like that, you can take it, guide from the forest department, then you can go inside. I mean, it's a better side actually, because of everywhere there are so many smugglers there inside the forest, because we encountered several times. Because I worked as a biologist, so that we have always forest department support. So we're going inside, we're collecting the data and everything. If any researchers want to do research in the Statistical Bias research, so don't go alone, you can take it forest department, the support and a guide, then you can go, mean it's a better thing, sir. So you can see this is my conclusion. Because of I am an independent researcher, I am not getting any funding from any agencies. So I worked as a biologist. Department asked to do for inventory. So myself, I started things, and within almost 3 to 4 years, we recorded almost 2 species of new record, 2 species of new locality record, 2 species of rediscovery, and first report in India, and also the first report in South India, and one new species is Cycas Jadeja. So This is for, I am said actually, this is, I am not taking any guidelines from any professor or any of our scientists. It is my own interest. It is, as a biologist, I started. So really, I put my hard work, I got this much a result. And based on the proper checklist, I submit to government. So if the checklist also might be having some problems, maybe having some mistake. So please, you can verify it and you can rectify it for through your research studies or something like that. Because the ISER is doing for excellent studies, especially the Eastern Ghats, and they're doing for the all of the field work. My professor, Dr. M.C. Satyanarayana say, so they all are doing for the dead zoology. They're not studying for the live zoology. So now the ISER doing for the live zoology, live wildlife. Really, I feel very happy that because my independent, I done this much only, but I feel very happy to do that. But I do not get a big opportunity to do that. So that's what I done for my own interest doing there and here for preparing checklists and based on this data I'm collecting and publishing something like that. So this is based on the during our study, what are the species we encountered, those species published in the different journals. And recently also we published, especially for the Journal of Animal Diversity for Herpetology Diversity in the Central Eastern Ghats. So this much only done on my contribution. So I plan to do for more if I get good opportunity or collaboration. So I am here basically wildlife background. My family are the artist background, so that I mingle with my art as well as my subject. I'm making for the different kind of sculptures and interpretation center, nature walk, nature trail, landscaping through wildlife aspect to conserve biodiversity. There are so many projects, almost 75 projects of landscaping we done through wildlife to promote and conserve the wildlife specimen, so that my subject, my art is mingled and with you are doing for this kind of activities. So I would like to thank especially for the Forest Department giving this wonderful opportunity to work inside the forest. Also Conservator of Forest, Wildlife Management Circle, Tirupati, DFO, Assistant Conservator of Forest Biolab, and also my research team as well as Forest Department staff. So also all the support given by the Forest Department. I thank you so much for this golden opportunity. Thank you. Because a very short time, so that I finished as soon as, because we got only for 15 minutes. So thank you so much for this golden opportunity. So I want to present and what I done there for especially for the checklist and everything. So that much only, sir. Thank you. Thank you so much.
Thank you so much. Do we have any questions? In case of no questions, we move forward. Thank you, Dr. Bhupesh Gupta.
Thank you.
Our next speaker is Dr. Vikram Aditya. He is a postdoctoral research associate at A3. He'll be joining us online.
Thank you, everybody. I'm going to share my screen now. Is my screen visible?
Hello. Yes.
Yes,
we
visible.
can see you.
Okay, so I'll start. Good morning, everybody. Good afternoon, rather. My name is Vikram Aditya. I currently work as a postdoctoral research associate in A3 Bangalore. My talk is titled Mammals in the Northern Eastern Ghats, the Present and the Future. I'm going to present some results from my PhD work starting from 2014 and the subsequent work that I've been doing in the Northern Eastern Ghats landscape till 2020. To begin with, as I'm sure most of us realize, mammals are among the most threatened of taxa. Over a quarter of mammal species globally are facing extinction, and over 50% of mammal species are facing population declines rapidly. When it comes to India, over 20% of large mammals are currently faced with extinction, and many have disappeared from greater than 90% of their home ranges. In this context of, I'm sorry.
Sorry to disturb you. Can you make your slides, make the presentation into a slideshow?
Sure. Is it in slideshow mode now?
Yes. Thank you so much. Sorry to interrupt.
No. Yeah, so to continue, in this context of mammal extinction, the role of neglected landscapes like the Eastern Ghats landscape broadly, and particularly the role of specific protected areas within them in supporting mammal populations is not well understood. And therefore, identifying landscape characteristics that influence mammal presence is critical for their conservation, particularly in landscapes like the Eastern Ghats, where very few studies have been done. So in this context, I started my PhD work in the year 2014. I wanted to look at mammal diversity and distribution patterns in the Northern Eastern Ghats landscape, which is the highlighted portion here in yellow. The Northern Eastern Ghats spans the states Andhra Pradesh, Orissa, and a bit of Telangana. The Eastern Ghats, as most of you realize, is a 1,600 kilometer long hill chain. It runs parallel to the east coast of India, starting from Orissa till Tamil Nadu. The Northern Eastern Ghats is inhabited by several different tribal communities, largely the Koyas and Kondareddis, but also smaller tribal groups like the Nayakpots, the Konds, the Savaras, Parajas, Gadabas, etc. The Northern Eastern Ghats supports a diverse set of flora and fauna. Unfortunately, the region has experienced decades of armed conflict between insurgent groups and security forces. The Northern Eastern Ghats also has is considered by some to have the lowest percentage of protected area coverage among forest landscapes in India. Only about 3 to 3.5% of the Northern Eastern Ghats is protected in some kind of a protected area type, either be it a wildlife sanctuary or a national park. The region has also experienced decades of land cover change, largely as a result of dams, shifting cultivation and plantations. Several dams have also come up in the region which have resulted in forest conversion. So this is the region where I started working in 2013, 2014, the Papikonda National Park, as you can see here. I also worked in the buffer of the national park. And over the past 4 to 5 years, I've been working largely in the larger Northern Eastern Ghats landscape, both in Andhra, which is the highlighted region here, and also a bit in Orissa and Chhattisgarh. To tell you a little bit about Papikonda National Park, this is one of the oldest protected areas in Andhra Pradesh. It covers an area of 1,012 square kilometers. It has quite a large elevation range, starting from 20 meters at the Godavari River. The blue region that you see is the Godavari River. Like many of the others have also mentioned, this is a major biogeographic divide for several species in the Eastern Ghats. The Godavari cuts across the Papikonda National Park from west to east. And that's where the lowest elevation forests of the Northern Eastern Ghats are there. But from there, the topography climbs rapidly and it goes up to 800 meters. The national park is located in the east and west Godavari districts of Andhra Pradesh. Like I mentioned before, it was first declared as a wildlife sanctuary in 1978. And in 2008, it became a national park. The vegetation is mostly moist deciduous type, but there's also patches of semi-evergreen forest and dry deciduous forest, and a bit of scrub vegetation along the fringes of the landscape. This is what the region looks like. The first photo on the left is the Godavari River, which bisects the national park into north and southern halves. And along the Godavari River, which is the lowest elevation regions, is mostly dominated by dry deciduous forests. But as you start climbing up, once you go beyond 100 meters, 200 meters in elevation, you rapidly encounter moist deciduous forests. And the higher regions of the national park and going into the larger Northern Eastern Ghats beyond that is mostly dominated by moist deciduous forest, and there's also patches of evergreen vegetation there. Unfortunately, the region has experienced a rapid land cover change in the past 3 decades. Several dams have come up. During the course of my PhD, starting from 2013 to 2016, 4 dams were either in the process of being built or had been completed, were completely built by that time. There's only one large highway passing through the national park. This was a single track before, and it was made into a double track highway, the State Highway 41. Several lift irrigation schemes have also come up in the Northern Eastern Ghats, and these are pipelines being laid to carry water from the lift irrigation schemes. And shifting cultivation used to be very rampant in the landscape before, and this involves clearing of large areas of forest and then burning the vegetation and then planting in these regions. The region has experienced a lot of land cover change, like I mentioned before. This is from my PhD work. So between 1991, which is the earliest available satellite imagery of the region, and 2014, when I did my PhD, I found that 102 square kilometers of forest had become degraded. So the canopy had reduced in this time period. Whereas in the regions around the national park, much larger area became converted from forest to non-forest in an area of about 490 square kilometers. And this work was published in the journal Ambio in the year 2019. These portions that I've highlighted here are 4 dams that came up in the region. And as you can see, around the dams, there was a lot of forest conversion. So I wanted to examine how are occupancy and diversity patterns structured across different habitats in the northern Eastern Ghats landscape, and how did protection in the form of the Papikonda National Park affect diversity patterns in the region? What habitat types were most influential in driving patterns in diversity and occupancy? This part of my work was published in different journals, mainly the Current Science, and a paper also got accepted in Biotropica this year. So to study mammal diversity patterns in the Eastern Ghats, I employed mainly camera traps because mammal densities are unfortunately very low in the landscape, and camera traps are the only way to go if you want to study mammals in the wild, particularly in the northern Eastern Ghats. So I set up camera traps in about close to 300 locations in the national park and outside the national park in the buffer. I also did sign surveys of hoof prints, bug marks, and of scats. And in the regions where I set camera traps, I also quantified the habitat structure. So this included counting the number of trees, counting the number of shrubs, measuring exposed soil surface, and also looking at distance from the camera trap stations to the nearest deforested patch, nearest village, as an indicator of human disturbance in the region. So some of my results, I recorded 28 species of mammals from my camera traps, and I enumerated about 55 species totally from interviews with local communities, tribal groups, and also from literature surveys. I found that the northern Eastern Ghats mammal composition was heavily uneven. It was mainly dominated by wild boars and barking deers. But in recent years, the work that I've been doing, my long-term monitoring that I've been doing after my PhD till the year 2020. So I also found that in moist deciduous forests in higher elevations, there's also some carnivore presence. So I recorded tiger and leopard. We've been monitoring 2 leopards that are moving around in the areas immediately close to our field station in the Chintur region. So I found that the habitat type, presence of water, a higher elevation, a higher NDVI, which indicates a higher forest density, as well as tree cover, litter depth, and the presence of the national park. All these factors positively influenced mammal diversity. So mammal diversity was greater in these regions, which had higher NDVI forest density, which were at a higher elevation. So basically to say that moist deciduous forests had a higher mammal diversity than dry deciduous forests. The presence of humans and domestic animals, and the decrease in forest NDVI between 1990 and 2014, these tended to negatively affect mammal diversity. Particularly herbivore diversity was positively affected by the presence of a higher tree cover and a higher NDVI. And I found that large mammals, including the sambar and the gaur, they were mostly present only inside the national park. So to conclude this part of my presentation, I detected 28 species directly from my camera traps. These included first records of species like the leopard cat, the rusty spotted cat, the honey badger, and the smooth-coated otter. Mammal diversity and abundance was much higher in the moist deciduous forests, which are also at the higher elevations of the northern Eastern Ghats landscape. Several rare species were also found, but these occurred only inside the national park, and I did not detect these outside the national park. But this changed in the work that I've been carrying out after my PhD till about 2020. And in fact, I found honey badgers and leopard cats outside the national park as well. Unfortunately, the northern eastern Ghats has very low detection frequency of animals and very low captures in camera traps when compared to other landscapes that also have been conducting small projects like the Western Ghats in KMPR, for example. Large mammals were largely present mostly inside the national park. And I found that large herbivores like gaur, sambar, and four-horned antelope, and also rare species like pangolins were particularly at risk. And this is to go on to talk about hunting, which I'll be talking now, which I realized is a very important driver of mammal densities in the Eastern Ghats, and it's probably the one and only major threat for mammals in this landscape. These are some of the camera trap images from my PhD fieldwork and the subsequent work that I've been carrying out. The sloth bear, wild boar, tiger, and small Indian civet. This is the leopard cat, leopards that we've been encountering quite frequently nowadays. Gray mongoose, honey badger. There are some herbivores. So during my PhD, I also realized that hunting by local communities is a very important driver of the lower numbers of mammals that are present in the northern eastern Ghats. And this is probably a major issue to be addressed for conservation in general in the landscape. So I did a simple comparison of species that are caught by communities. I interviewed about 60 local community members for this work, almost all of whom were hunters. So I found that the species which are most abundant that I recorded in my camera traps are also the most frequently caught by communities. So this includes species like the barking deer and the wild boar. However, species so the monitor lizard and the giant squirrel were also caught, which were not mammal. The monitor lizard is not a mammal, but these were also caught. However, I noticed that some species which are preferred for hunting by the local communities are not frequently recorded. So species like the four-horned antelope and the porcupine, these are preferred by communities for their meat as a delicacy, but these are not very frequently caught. For the past 3 to 4 years, I've been looking more at the impact of hunting and illegal trade in wildlife on mammals in the northern eastern Ghats landscape. Particularly, I've been interested in the impact that it has on the pangolins. The pangolins, as most of you may know, there are 8 species globally, 4 in Africa and 4 in Asia. They are the most highly trafficked group of animals in the world in illegal wildlife trade. In India, we have 2 species, the Indian pangolin and the Chinese pangolin. The Chinese pangolin is a critically endangered species and the Indian pangolin is endangered. And both of their populations have rapidly declined in the past 2 decades. This is despite them being protected nationally under the Wildlife Protection Act as well as internationally under the CITES Convention on international trade in wildlife. Several reports have indicated that the numbers have declined rapidly. A report by Traffic India in 2018 said that over 6,000 pangolins were seized in illegal traffic between 2009 and 2017. A report released this year only by CITES says that more than 1 million pangolins have been estimated to be caught in illegal traffic in the past decade, most of this originating from Africa. So like I said, there are 4 species in Asia and 4 in Africa. There are 2 in India. The Indian pangolin is distributed across peninsular India, whereas the Chinese pangolin is present across the northeast of India as well as the Terai Arc. Pangolins have been in the news recently because of their. There were some studies which claimed that the Malayan pangolin could have been a possible reservoir for the SARS-CoV-2 virus, which causes COVID-19. However, this has not been proved yet because the pangolin, the studies which said this have only been able to access to retrieve the coronavirus from illegally seized pangolins, whereas the wild pangolins, the Malayan pangolins which are in the wild, do not have this coronavirus. For this work in the northern eastern Ghats, we sampled these grids. We set up camera traps close to burrows and anthills and termite mounds to detect the presence of pangolins. In these grids, we also interviewed villages that were present in these grids for gathering local community knowledge on pangolin presence. We found that the pangolins, they prefer a huge variety of habitats. They're not specifically restricted by any habitat as such, but they prefer steep boulders and steep slopes and dense vegetation habitats.
Hello.
They tend to burrow near boulders with multiple entrances. They also climb trees occasionally. Hunting in the Eastern Ghats peaks post monsoon. I'm sorry. Yeah.
Sorry to interrupt you, but we are overshooting the time. Can you please wind up in 1 minute or two?
Sure. I'll do that.
Thank you.
Yeah. Hunting is very rampant in the northern Eastern Ghats, particularly of endangered species like the pangolin. Pangolins were frequently caught earlier, but they've become very rare now. So catching of pangolins has also declined. However, when it is caught, the earlier times meat was used to be consumed, and scales were meat was consumed while the scales were discarded. But nowadays the scales are sold at huge prices, and people also tend to keep the pangolin alive and they try to sell it because now they've realized that there's a market for pangolins in illegal trade. Scales are used as a defense against black magic. Communities who believe in black magic use this, and they make rings out of the scales and they wear them because they believe that this defends them against black magic. This is how communities hunt in the northern Eastern Ghats. It's quite common, and it's common. Anybody who goes on the road can see people going around with bows and arrows. They also use wire traps and snares. These are larger snares. These are for larger animals like the barking deer and wild. Use of guns is very, very rare, but you do see them, especially if you set up camera traps. It's quite common to encounter hunters working with weapons. I'll skip this. We've been also monitoring hunting patterns in the northern Eastern Ghats and also trade, particularly of species like the pangolin. Pangolin hunting and trade in general spiked during the first phase of the lockdown in 2020. This spiked by almost 3 times when compared to the previous years. To conclude, hunting in the northern Eastern Ghats used to be largely opportunistic in earlier times, but nowadays it's becoming more targeted, especially of tried species like the pangolins. And communities have reported that pangolin numbers are very, very rare. The sightings are also very low due to the hunting pressure. This is what communities themselves say. What I feel is that regulating hunting and trade, especially locally, is key to conservation of wildlife, especially endangered species like the pangolins. This is some of our published work over the past 3 to 4 years. Yes, this is our published work in the past 3 years. I'd like to thank IC Tirupati for organizing the symposium. I think it's very, very important to have symposia like this for the Eastern Ghats, and for inviting me to for the talk. A3, Dr. Rajesh Mahapatra and the Nandankanan Zoological Park for their support in our pangolin and hunting work. The AP Forest Department for giving us permits right through from 2014 onwards. Several field assistants, friends and volunteers who've been part of our team. Interview respondents. And all these agencies for supporting our work. Thank you very much.
Thank you, Dr. Vikram. We'll take the questions in chat. We'll go ahead with the next speaker. Thank you so much. Next, we have Mr. Rajashekar Bandi. He's a Citizen Science Coordinator in IC Tirupati.
Hello, am I too loud? Okay. Yeah, very good afternoon. I'm not going to take much time, and today I'm going to. So it's a bit different from what my colleagues Archita and Chiti spoke about. So it's about understanding the avian diversity in the Eastern Ghats of Andhra Pradesh through detailed missiness-based resurveys and using the citizen science data. So we've been speaking about the Eastern Ghats since yesterday, and it's very much largely unexplored. And also the bird data from the landscape is very less when compared to the last 30 years. So the long-term surveys that happened about like 90 years ago in the landscape were never replicated. So there is a like very much of the protected areas in the landscape is unexplored. So before going further, sorry. So I will just quickly give a glimpse at what happened to the historical contributions from the Eastern Ghats landscape in the Andhra Pradesh. So T.C. Jadon, you don't need any introduction. So the Jadon's courser was named after him, and he worked out of Nellore, and he was a surgeon working with the British at the time. And he collected a lot of birds from the landscape, and he described a lot of birds. And that's in 1840s to 1850s. And after Jadon, the largest and the wide survey that happened in the Eastern Ghats is between 1929 and 1930. It was funded by A.S. Vernai. It's called the Vernai Survey of the Eastern Ghats. About 130 species were recorded. And after Vernai, Humayun Abdul Ali, representing BNHS, he collected more than 140 species. And he added 73 new species to the list of the species described by Vernai. And after 1940s, it's Trevor Price, who worked in the Northern Eastern Ghats region. And he was there for about 4 to 5 years and collected long-term data through mist netting. So if you look at it, Trevor was the only person who did the mist netting, and the other modes of collection is by shoot and collect, which was the traditional way of doing the avian assessment in those days. Just to add, there was one more person who was very little known from Andhra landscape, K.S.R. Krishnan Raju. And he did a lot of mist netting supporting the BNHS in the landscape. But of these, only Vernai was the only person who did the large-scale surveys. So right starting from the Seshachalam landscape or the Kadapa district, all the way to the Sankarmetta in the Northern Eastern Ghats in the Visakhapatnam district, the survey covered quite a large bit of area. But Abdul Ali, Trevor Price, their work was moreover restricted to Visakhapatnam Ghats only, but the Southern Eastern Ghats was never explored much. So this is the map of the Vernai expedition. You can see these black dots here. These are the locations where Vernai survey did the data collection against the birds. So up to the Visakhapatnam here, all the way till somewhere here. We are here in Tirupati. So they have surveyed multiple locations. So, what we did is we just want to see how much of these landscape was covered and explored in terms of avian data. So these yellow spots are the locations covered by Vernai during the survey in 1930s. And then we took the latest citizen science data from the eBird, about 5,000 locations, like the checklist locations were taken. And you can see most of the exploration of the avian life in the landscape was done along the roads where most of the places were accessible. And the protected areas which are marked are highlighted in green. They are largely unexplored. So this will give us a clear picture of, there are only some concentrated hotspots, like this is Tirupati, we have a large birding community. And Pulikat was visited by a lot of bird watchers, and then you can see a lot of data here. And again, the Koringa Wildlife Sanctuary, and then the Visakhapatnam has a Vizag Bird Watchers Society, a small community of bird watchers. So we have some little hotspots where the bird data is being generated, but most of the protected areas are widely unexplored. So what are the current gaps in the landscape? So there are no strategic explorations and surveys that happened in the past 90 years. And also the avian diversity of, most of the protected areas, as we said, is not explored. And we don't have enough data to conclude the status of the species from the landscape. And the last checklist of the Andhra Pradesh birds was done in 2013, before the bifurcation of the state. Now Andhra is split into Andhra and Telangana. So as I said, we recently worked on the new checklist of birds, and only the current Andhra Pradesh, we came out with about 498 species. And the data on the birds outside protected areas is not, is also not much, because we don't have big or larger birding communities in Andhra when compared to Kerala and Karnataka. So we want to fill these gaps. So we want to fill these gaps in a strategic way through citizen science and also by research. So I strongly believe in citizen science, not because I am the citizen science coordinator here, but I am from a non-science background, and 9 years ago, Dr. VBM Madhur, he's here. He took an initiative of a conservation course at WII for people with no science background, and I am one of the first persons who joined the course. And now after 9 years, I am standing here. So creating awareness and citizen science is a very strong tool, and it can make a lot of difference. So one of the ways we want to unearth or explore the avian data of the landscape is by citizen science. So we want to do capacity building on like taking people for bird watching, and then helping them in identifying the birds of the landscape, and also supporting them in recording and monitoring the birds, and also to create awareness, like why birds. So what are the ecosystem services? What are the importance of the birds and the conservation of the species? And also we are empowering them with the technology like eBird and iNaturalist. There are a lot of tools where people can actually upload that data and share with the wider communities. So we have a citizen science program at ISER Tirupati, and we have created a AP Birdwatchers group in 2019. And now we are fairly about 80 birdwatchers approximately. And also we've been supporting the local universities. In Tirupati, we helped in creating birdwatching clubs at the veterinary university. And we are taking a lot of people for birdwatching in the SV University campuses and the Padmavati University. So a lot of data is being generated from the Tirupati landscape, and we are trying to replicate it over the complete Andhra landscape. And recently we also trained the forest department people, about 180 officers in bird identification and recording the birds. So this is a summary since 2019 December. You can see we did a lot of outreach and capacity building in the landscape despite the challenge we faced by COVID. So we engaged about 692 school children and teachers, college students. And during the COVID times, we did the webinar on birds and biodiversity of the landscape with about 2,000 audience. And most important thing, we are now in terms of establishing a collaboration with the Andhra Pradesh State Forest Academy to develop a curriculum for the officers in training, so that we can impart knowledge of not only birds, but the identification and recording and monitoring of the biodiversity. And we also added 11 new species, birds, butterflies, moths, and everything together to the biodiversity of the Andhra Pradesh. These species were never recorded before. And then as a part of our citizen science and capacity building, we are also actively coordinating the global events like Great Backyard Bird Count. So through our birdwatching clubs and the AP Birdwatchers group, so we made a lot of difference. This is the data from 2007 Great Backyard Bird Count. You can see this is only Tirupati. There are a couple of checklists. So these red dots were all the checklists. And there are 2 or 3 checklists here. So fairly there was no birding happened during the Great Backyard Bird Count of like the 4-day event. And if you see in 2022, you can see all the 13 districts in the state of Andhra Pradesh have been covered. So birdwatchers from all the 13 districts contributed the data. So almost 2 to 3 checklists in 2017 to 100% data contribution from all the 13 districts is a monumental achievement for us. And also, not only as a group or as an institution, we are doing this, but we are also supporting the other institutions in the landscape. So this is the campus bird count. So you can see that between 2020 and 21, we have supported so many campuses to participate in the campus bird count. There is a 166% increase in the participating campuses in the landscape. So we've been supporting them with running the bird walks during the Great Backyard Bird Count. So through citizen science, what we have achieved so far is Dr. Bhupesh Gupta, during his work, he has recorded 215 species of birds from the Seshachalam landscape. But we recorded 218 just from our campus, which is 4 kilometers from here. And now the total species of birds recorded from the Seshachalam is 284. So that is a revised number. So, and we are right next to Wildlife Institute of India in terms of the checklist submitted during the Great Backyard Bird Count and the bird species recorded. So we always compete with them. It's a very healthy competition for us. And as I said earlier, from 0 checklist almost in 2016 to 100% participation in GBBC, this is a monumental achievement for us. And also currently we are working on Tirupati Bird Atlas project, creating a bird atlas for Tirupati, funded by National Geographic Society. And also through the community of the bird watchers across the Andhra in all the 13 districts, we are trying to generate the bird data outside the PAs. And we're also in the process of developing a handbook of birds of Tirupati, and a checklist of birds of Andhra Pradesh is almost completed. Hopefully we are going to come out with it this year. So with this, the citizen science part is completed. So as a part of it, we are supporting the forest department with the bird surveys and all. These are a couple of reports which we submitted. During the COVID time, we initiated the Vijayawada winter water bird census with the citizens of Vijayawada. So the first walk, when we started with the first water body, only 2 people turned up. But by the end of the census, we have 36 people. And this year, I couldn't lead it. I was training the forest department people. But now the citizens took it themselves and they have done it this year without our intervention. And also we made a checklist of the birds for the forest department during the training, and we have made a report. So it's not just the data we are using it for ourselves, we are generating it and sharing it with the wider communities and the forest department as well. And finally, the other component we are looking at is the research. So other than citizen science, we have a very good research capability. Dr. Robin is leading here, and we have a very strong research community at ICER in the Bird Lab. So with the help of our lab group, so we are trying to do strategic surveys. So we want to replicate the Verneuil resurveys backdated to 1930. We want to revisit the same locations because the landscape has changed, the forest has degraded, there are a lot of urbanization happening. So we want to do the resurveys, and also we want to do misnetting in the landscape to get the species richness, abundance, and the survival rate of the species. So just to give you an intro, the current work we are doing is the biogeography work, the misnetting work across all the 7 states in the subcontinent. And we have misnetted as a team more than 3,000 individual birds with a vast experience. And the misnets are very much safe, and they don't, normally hurt the birds. And over 900,000 birds are ringed every year in Britain and Ireland alone. So to conclude, we want to combine our capacities and the strengths. So we have a vast strength and experience in the citizen science, and combined it with the research capability we have. We want to generate a long-term bird data from the landscape, and we want to explore the landscape widely, not only outside the PAs, but within the PAs. And on the citizen science front, we are well ahead. We are doing very well with a lot of collaborations with more than 14 institutions in the landscape. But on the research bit, we need a lot of support from the local forest department and the biodiversity board, probably. And we are getting funded by Dilip Matai Nature Conservation Trust, who is our major supporter of the Citizen Science Program. And we have multiple grants from National Geographic Society, Cornell University, and IIT Tirupati are our very big supporters. And of course, MOEF and SERB. So the only help we required in the landscape to initiate all these things is permits. Again, it all boils down to permits. And as my colleague Archita mentioned earlier, we have everything except permits. So I request the Forest Department personnel and the State Biodiversity Authority and the National Biodiversity Authority to kindly look into this and do the needful for us. And hopefully in the coming years, we hope the citizen science will prosper and we see more results from the landscape. Okay, thank you. And I thank all my colleagues here and Nandini, Robin, and Dr. V.B. Madhur, and all the attendees. Okay, thank you and have a nice afternoon.
Thank you, Raja. We have our next speaker, Dr. Ashish Jha. He is a postdoctoral fellow from IISER Mohali and will be joining us online.
Hello everyone, I'm Ashish Jha. I'm currently working as postdoc at IISER Mohali, and I'll be presenting the work I did during my PhD. So my association with Eastern Ghats is because of this species called yellow-throated bulbul. And for this species, I studied its biogeography, population genetics, environmental niche, natural history, all part of my PhD. A brief about the species. So it was described by T.C. Jordan in 1842, and this is endemic to the Deccan Peninsula. So the green region you see here, that is Deccan Peninsula, and the red spots on these green regions, those are independent and isolated occurrence records available for the species. The species is described, is classified as vulnerable in IUCN Red List, and the prime reason is the habitat loss. So these are few of the threats to its habitat. So this is mining of soil, lopping of trees. So these are ficus trees which grows out of rock, and they are very prime source of nutrition and food for the species which are found in these kind of landscape, primarily yellow-throated bulbul, and obviously mining for these rocks. So these are granitic rocks, and India is one of the leading exporters of granite, and the habitat of yellow-throated bulbul dispersed across peninsular India, they hold up to 70% of granite reserves of the country. So it's a very lucrative for the government to mine these and export as a source of revenue. So that is one of the biggest threat for the species. This is found in rugged terrain, and like all other bulbul, this is also an omnivore. It feeds primarily on fruits and berries and also on insects. So these are the 4 different kinds of habitats where I have recorded the species, and you can see it varies from almost no canopy cover in Hampi to very high canopy cover and very moist forest in Ahobilam. But whatever be the area, one of the key feature is the rugged terrain and these exposed granitic rocks. Now there are these mesic patches or moist or wet areas created by the sipping or this dripping of water, underground water, which sips through these rock crevices, and it attracts lots of birds. And YTB is associated with this sort of places, and these sort of places are also places of pilgrimage and worship. So in an otherwise very dry landscape, these are like moist oases. So I felt that probably this could be one of the variables dictating the distribution of yellow-throated bulbul. So I did a species distribution modeling exercise where I used this as one of the variables. To calculate this as a variable, I got NDVI layer, the vegetation index, and I created, analyzed its coefficient of variation. So if the variation in vegetation index is high, say during monsoon and non-monsoon period, then it means that during monsoon it's very wet and otherwise it becomes very dry. But if there is these sort of water sources, the vegetation would remain green throughout the year. So the coefficient of variation would be low in such areas. So this is the outcome of the species distribution modeling. I did it for the peninsular India, wherever the species is found. So these green areas you see, these are the distribution, the predicted distribution of the species. Red regions which you spot here, these are the shapefiles for protected areas in this region. So currently about 12% of the predicted distribution falls within the protected areas. Among the variables, the key variables were precipitation of the wettest month. So the species is found in Western Ghats, but only in the eastern slopes. The dry, the leeward side, not the windward side. So the species clearly avoids regions of high precipitation. Okay, then if it's found in Eastern Ghats, which can go, the temperature can rise very high during the summer. So I would assume that this prefers drier areas, but it's not so. So the next variable is a mean temperature of warmest quarter. So we see as the mean temperature for the warmest quarter or warmest month increases, its probability of occurrence goes down. So it certainly does not prefer very dry or very warm areas either. And with the coefficient of variation, yes, as the coefficient of variation increases, its probability of occurrence goes down. So it prefers areas which remain green throughout the year, which do not receive a lot of rainfall, and certainly do not have very high temperature during the summer months. So I feel it's probably these rocks, these boulders, which provide some sort of microclimate to the species and protection against the harsh weather, which allows the species to persist in an area. But no such study has been done. So this is something which I look forward to doing in future. The next thing I did for the species was to create a phylogenetic tree and to understand the sister species and how did it evolve. So when I started this work, there were bulbul phylogenies available, but the Indian species were missing from the global phylogenetic tree. So I collaborated with people in scientists in the country and in Sri Lanka, and I got the samples of Indian species. So in India, we have 24 different bulbul species, and 12 of them are endemic or near endemic. So India has very high diversity of bulbuls. Here I'm showing the phylogenetic tree only for Pycnonotus, and we see that our Pycnonotus xantholemus, this does not have a sister species, and it evolved. It's one of the oldest lineages which diverged around 7-8 million years ago. Another endemic species of the same habitat is white broad bulbul, Pycnonotus luteolus, and that too is an old lineage, but it diverged around 6 million years ago. Now, this was quite surprising because the species are endemic to dry areas. And we know that once upon a time, entire peninsular India was covered under moist forest. And as aridification happened during Miocene period, these dry habitats became available. So I would have expected the species adapted or species endemic to dry region to be newer than the species in the moist areas, but that was not the case. So next I wanted to look into its population genetics. So this species I sampled in around 30 different locations, and you can see that there were few areas where I could not capture the species. So in all, I captured around 63 individuals via misnetting from 20 different locations. And I created a haplotype network. This was done using the control region, about 1050 base pairs. And what I found was that various populations from different regions, be it the northernmost population in Uma Maheshwaram, Achampet, versus the southernmost population in Udumalpet, Tirupur district of Tamil Nadu, the species share haplotypes. So it looks like they all are genetically connected. There's another study that I did, which showed that species showed signature of demographic expansion. Now, that was quite surprising. But one of the reasons which I hypothesized could be this, So as the species must have evolved somewhere in southern India as the identification happened, and with the gradual identification, more and more habitat became available. So the species expanded its range, and species, that's how shows signature of demographic expansion. Now, another test of this hypothesis would be to do the same analysis for another endemic species such as our white-browed bulbul, and not just birds. We have endemic geckos of the same kind of habitats. So doing some sort of population genetic study on them, and if you find signature of demographic expansion, then it validates this hypothesis. It also appears that the species is able to move across habitat. Now, we know that this is a rare species. Even in the habitats where it's found, I have not recorded more than 5 individuals. And at most, I believe there might be 10 breeding pair in one location. There has been no such focused life history or natural history study on the species, so we cannot give exact numbers. But this is what I estimate, that in a given habitat, it may not be more than 10 breeding pairs. And that is a good recipe for local extinction, stochastic extinction. So how to avert that? The only way a species can avert local extinction is via dispersal. So it looks like the species disperses from one habitat to another, but we have not seen it, we have not recorded it, because there has been no long-term monitoring study on the species, no ringing studies, or no telemetry studies. But there are anecdotal records which I compiled from various literature sources, as well as through personal communication. For example, there's a publication in 1993 which talks about how the species moved into Rishi Valley campus in Horsley Hills after the greening of the area. But Horsley Hills is just connected to the Rishi Valley. So it's not a big surprise that the species was able to disperse into the campus. Then there is this campus called Campus of Learning, Center for Learning, CFL, which is about 4-5 kilometers from Savandurga. And one of the faculties at CFL told me that the species visits their campus only during summer months. So there are such anecdotal records of species moving out of its prime habitat during pinch period, during summer periods. And that could be the reason why the species shows a panmictic population structure. Now, this is a very specific kind of habitat called granitic inselberg or inland hillocks. Now, as I said, about 12% of its predicted distribution is under protection. But what about the remaining 88%? And in fact, about 80% of its occurrence records came from these inland hillocks. And they fall outside protected areas, and they are rich source of granite. So it makes them highly vulnerable to habitat exploitation. So I propose that yellow-throated bulbul can be declared as a flagship species for protection and conservation of these inland hillocks. There are no other major species such as bear or leopard, or certainly not tigers and elephants in these habitats. And these ficus trees which grow out of these rock crevices, they may take up to 10 years to come into fruiting, and they are prime resource for food for these species and for yellow-throated bulbul and all other species which thrive in these habitats. So for protection of these kind of unique ecosystem, we need a flagship species, and yellow-throated bulbul can be one of them. So if we, I'm ready to take questions, and here my presentation ends. Thank you.
Thank you, Dr. Ashish. Thank you, Dr. Ashish. Do we have any questions? There are no questions. Thank you again. So we'll break to the lunch for the lunch break. We are ending the section. I'll hand over to Dr. Nandini.
Thank you, everyone, for staying on well past 1 o'clock. Thanks to all the speakers who joined us virtually. It was great to have you be part of the symposium, even though many of you couldn't make it in person. Can I ask Dr. V.B. Mathur to say a few words to close the symposium? We're also extremely grateful to Dr. Mathur. We wouldn't be here without him. This entire symposium is his initiative, so we're really grateful.
Well, thank you so much. I must say that the last 2 days have been very fascinating. Not that I had any doubt about that, when we were planning with Dr. Nandini, we did say that I did say to her that if you do it on a weekend, I will be there. And yesterday, of course, we had a different flavor. Most of you know we had presentations looking at the ecology, biology, and related issues with red senders, which happens to be a very important species for the Eastern Ghats. And after this discussions, we had our internal meetings also with the PIs. And I emphasized that, see, they are, you saw those presentations. Those presentations are being made or have been made on certain identified topics which they conceived 2 or 3 years ago. And then we provided funds, and they are using the students and researchers and faculty. But I am very clear that research on red centers cannot just stop by those 6 projects alone. And that is what I emphasized in our evening meeting, that whatever new questions which are coming up and new ideas which are coming up, and there are further gaps which are being proposed, those need to be also addressed and fixed. And obviously those institutions and PIs who have spent their time and effort in reaching that have, of course, the first idea of mounting second phase or third phase of the projects. But I did emphasize that if a quality manpower is also available at Tirupati, either in ISR or in IIT or any campus, those should also be encouraged. So we have suggested those ideas. So there will be opportunities coming up, those who are following it, those who have interest. And we also need to look at, while we say it's research coming under red centers, you understand that a range of species are getting protected. What we now understand as a keystone or an umbrella species, there would be opportunities there also. So that was the first part I wanted to share with you. But the important part is now what I'm going to say. I had a good fortune of talking to both the directors of the IIT yesterday and ISER today. And of course, Professor Ramesh as well. See, we need to change the way in which we see the growth of science. The ISER campuses themselves have been set up with a certain purpose. Otherwise, we have universities which had zoology, botany, chemistry. I myself studied, and many of senior people like you would have also studied. But they are not delivering, or they have not delivered. And that is where the ISER system has been conceived, where you bring about integration. All kinds of basic sciences are being taught. And that's what my point was. And then I'm sure you know, this point was made to me yesterday only, that Tirupati is the only place in India which has an IIT, which also has an ISER. See, we need to make use of this kind of convergence which is there. And today's modern science cannot grow without input from whether you are an engineer or a technologist or an IT person, or let's not forget the use of artificial intelligence. See, all these things, the data that you are trying to generate, imagine you have been talking about citizen science. See, you yourself said 3,000 records against 9 lakh records in UK and Ireland. So we have a long way to travel, but we need to prepare ourselves that as the data increases, as the big data is there, what kind of capability we need to develop that. So just to keep it short, we did discuss with these 2 directors, Dr. Nandini as well. And there is an understanding that we need to increase the way which we have been dealing with, whether you call it forest, whether you call it environment, whether you call it biodiversity conservation, or I would say nature conservation. And you have lovely campuses. Lots of things can be done on campus itself. Believe me when I say my own understanding of my own campus in WII, we have about 300 acres of land. That land, that campus was very, very important for all things. As they say, charity begins at home. Everybody who comes to stay in the campus in the morning, he is there on the trail. So we need to look at that mechanism. I understand they're already there, but we can formalize those kind of a thing so that a regular interest. See, on campus activity, there can never be a dull day over there. So those are the kind of things which we need to encourage. And those who have an ability to go out in the forest, identify birds, mammals, whatever, should take the lead in that. And it's only the young student who joins at year 1 has come from wherever in the country, but has no idea what nature is. And that is what we need to do. So we have to deal with 2 campuses. We will be, this symposium of ours is a beginning. I don't want to end it here. We need to have some more plans that we can have it. The director, ISER, has already said that he wants plant sciences or biological sciences to come up as a major focus. And this ISER to be recognized. See, every institute can have its brand value for something. And that he has understood it and he has himself communicated. So we should be able to work out some more things so that there are more opportunities and there are more activities and there is not a dull day on the campus, no matter what the temperature is. So for every hour of the day, there could be something over there. Today morning was very nice. I had a flavor of seeing a large amount of research. As I said yesterday, I did say that there is not much research in Eastern Ghats, but I see today people with PhDs on species in Eastern Ghats. So obviously there is traction. We need to scale it up, and all kinds of studies are being talked about, including camera trapping, including citizen science. We need to move ahead on them as well. But the last thing I want to say, which is very important, I am very happy that my colleague, Mr. Reddy, who is the chair of the Andhra Pradesh Biodiversity Board, is here. See, sir, this morning we heard several presenters. Everybody had an issue. Not that you and I can solve it immediately, but I must put it across that permissions. See, these permissions, that's why when I scribble in my notes, I wrote a word called the dynamics of permission. Yeah, you must hear me now on this dynamics of permission. I will be very candid on that. See, same group, there was a person who said 4 years of wait for getting a permission. And then there were people who were saying everything is fine. So you can imagine that there are more than what you see when you come to the permission. If you permit me to be more candid, I would say, see, whenever it's a human nature first, everything which you and I do in our home or in my office, there has to be some element of incentive in it. Okay. Now you as a researcher, you know your incentive. That you want to study, you want permissions, you want to observe, you want to publish. Very, very clear which is there. But if I am the DFO, if I am the conservator, what is my incentive in your research? Okay. Now those can vary. It could be a very simple thing of writing a 1-line acknowledgement that this particular report or study was supported by that. If the person shows some interest, you can offer. I'm not saying, don't quote me out of context that I'm saying offer authorship to people who have not done anything. But you can create that kind of a thing. You can encourage them. You can ask them that, sir, it is there. And the simplest thing, this morning I had a discussion with the DFO here. He was saying that, and he was pointing out to ISER people, no problem with ISER as such. So he was saying that we work with them. But he had a point. He said that I have a staff. I have my rangers, I have my guards, I have my frontline staff, I have my contract people. All I want is that those people can also be trained. See, which is the simplest thing. Even our 1-year-old JRF can also talk about his own research or her own research and do that. So the point I'm making it that we also should also offer certain things from our side that makes the process easy. Okay. And it is nothing, it doesn't cost you. Every morning you can just offer, sir, I am going on my transact, I am going in that particular area. Please send your people along with you. So while you are doing your thing, he could be an extra help. Because see, what happens is sometimes there may be people in the department at the lower level who might be actually having more knowledge what you are having at this point of time. Over the years, when you acquire your degrees, you may become super kings over there. But at this point, so what is the feeling? that we provide you help, we provide you support, we provide, but in return, we don't get anything. So I'm just making a small suggestion that these are not something that any incentive which you need to bring or give something. It is only encouraging them. And those who want to be with you, those who want to acquire knowledge will come to you. And this message will go back that this is what we went to that group and we learned 3 new things or 2 new things. We got a new species. And if there is a new find, then it goes over there and somebody gets a little incentive or recognized. So that is what I would say, whatever you treat me as a friend or whatever, but to see to it that we also provide an extra bit on that, and which is not very difficult. What is there to hold a 1-day session or a 2-hour session on saying that, okay, this is it. You can tell them that, okay, and I told him, the DFO, that when you are giving a permission, you can also write 2 lines in that conduct a monthly program for my staff. What is there? There's no big deal we are asking. So we need to look into that. And of course, now that you have pointed it out to me, we will also talk, and Mr. Reddy is already there. We can also mark a copy, and sir, you can also look into it. See if there are procedures on that. The other complaint I can tell you, and this is not related to ICER or this part. As Director WI, I am saying, everybody tells me, if I can say first in Hindi, sir, aapka report nahi milta. We don't get your report. Okay. So, which is a point. Now what to do with that kind of a thing? We keep encouraging them. Fortunately, IT and websites are good. You can always upload your report and do that. Whenever you send to the department, now I'm saying the other side, you send a nice fancy report, he sees it, he says, "Acha, main isko ghar le jata. I will take it to my home." Then it doesn't come back to the office. Next man comes, he asks, "What is this project?" Nobody is able to tell, and the same story repeats that we have not been provided. So there are issues on both the sides, but we need to be a little flexible. We need to be a little, what should I say, a little more forthcoming and saying that if it is there, you can always say that this is my, even if I have done a literature search. Today, everybody is operating on an email. What prevents a student when you go over there or your PI goes and says that, okay, right now we are starting, we have just a literature survey, will you be keen? Can you give me an email? I can send it to you. So see, those kind of very small steps, but they create a different mindset over there. And then of course, if now increasingly the whole issue, because there is now in many states a CC of research, who is supposed to regulate research for the entire thing. And they are also creating expert committees. Now, when you create these bodies, when you create this position, there is some work to be done. So when we ask them what is happening, no, sir, we want them to make a presentation. So even making presentation is also no big deal for the researcher or the scientist. You can only say that, give me the time, give me the opportunity, you do that. And nowadays, even virtually, you can say that, yes, if you want to hear all those issues which, see, 5 years ago, 10 years ago, 15 years ago, getting permission for camera trapping was the most difficult thing. And those of you who know Dr. Ullas Karant and his story, he knows that in Karnataka for many, many years, he was just being deprived of that. So those days were different, thanks to WII, WWF and all those agencies. And I was so happy to see everybody is doing camera trapping. Now there is no mystery behind that. Everybody was earlier feeling if you put a camera around the tiger, the behavior might change. Or you might be introducing something for which the DFO will be responsible. So those days are all gone. But still, if any clarification is required, if any more communication is required, most of the time it is communication. Nothing else. That's why I said that whatever letters you have written, if you mark a copy to me, I will take cognizance. My only way of knowing what is happening in my organization or in this world is through a mail only. If I read it, I feel if any intervention is to be done, I just do it on the spot on that. So I'm not saying that there is one fixed, even if we say, even at WII, we were not able to do. All my colleagues said that there should be an advisory from the Ministry of Environment and Forest that WII report, research and researchers should be supported. Who cares? You can get any advisory done. See, these are all very, very small interpersonal matters. Nothing more than that. And the officer is also morally and dutifully bound to issue that. It's only that when he gets or she gets upset, then they say, this has not happened, this has not happened, we did not get. If you have given the report, then he'll say in 1978, there was a study for which I have not been given report. So there are higher ways of doing things, and there are 10,000 ways of not doing things. We need to work somewhere in between and see to it that how we can provide that. I'm not saying you could do anything extra or anything out of this thing, but just while you talk, while you communicate, while you do that, that is what we are doing. And those of you who actually stay and work inside the PA, see many of in WI, we have field stations everywhere. What happens? Somebody is coming. For the officer, I'm now pleading for the DFO. For him, information is important. If somebody is visiting, he will like to know what has happened, where, what has been cited, what has not been cited over there. You may say that you will share that information when you publish. And you can well imagine that the day you make your observation and the day you publish, I won't guess how many days will pass in between. Okay? And it might not even get published also. So the point is that interim manner, whatever it is, on some other day, Nandini, I'll speak on data paper. That's another thing which all my biologists need to understand, that while you must produce an academic paper from wherever you want to do it, there is also a possibility of what is now being called as a data paper. If you have just produced a checklist of birds or butterflies or ants or whatever, you can publish that kind of a checklist, which will be acknowledged against your name. If you go to the GBIF website, the Global Biodiversity Information System website, you see the big papers, Pentasoft and others. I myself have published a paper on that just to encourage. And what is the way that that data comes out? You need to curate a little data. You just can't just publish whatever you have handwritten. A little more information might be required over there. But that data paper, A, can do two things. One, it can acknowledge everybody, from the field assistant who carried your bag or the driver who took you in that vehicle. You can acknowledge that. There is no limitation on that. So even the driver will feel happy that, look, my name appears somewhere over there. That's one part. And you will be given a DOI. So that means that record goes against your name. Okay, and same kind of intellectual, what is it called, property recognition, or your name against that data is registered. Then you take your time, 1 year, 2 year, 3 year, whatever. And then when you produce your academic paper, you just quote it from that data paper on that. So see, this is what I wanted to do for long at WII. Sorry, I could not do that. Everybody has an apprehension that my data, I mean, you know how data is treated by people. And sometimes I say, if you permit me, people protect their data more than what they protect their daughters. So that is the kind of mindset we have. So in a modern day, neither the daughters need protection nor the data needs that kind of a protection over there. We need to come out. We need to say that, yes, this is it. And this is being published. And see, this one more thing which we need to understand, the progress in molecular science vis-a-vis the ecological science. What is the difference? In a molecular science, in a molecular lab, the moment you work out a sequence, you rush out and publish that. Why? Because then nobody else in the world will work on that particular segment for which a human genome sequence has been uploaded. What we do in ecological science, we just hold everything. Even if there is a change, you see, 3 times in the morning I said that climate change effects are there. Maybe your research can provide some pointers to that. But when you publish it, when you make it known, if somebody is now looking at, see, you remember the story, sparrow populations are declining, this is declining, that is declining. Where is the evidence and where is the publication? You don't have to wait for Trevor Price to come in and say that this is what is the data for the Eastern Ghats or for the Western Ghats or for the Eastern Himalayas over there. So the point is that we need to introspect, we need to look at it, modern technology, modern science, modern IT tools allow you to do that. Every sharing will be at your terms and conditions, but make it a point that you share it with you. Because if you don't share, then all the effort that we are doing doesn't come out in the open. Science does not benefit. Society certainly cannot benefit if science is not being shared enough. So with these words, I thank you once again. Profusely want to thank both Nandini and Robin, who have been instrumental in getting me over here. They've spent their time. But I want to assure you that this is not a one-off event. It may have started like that, but we are, as I said, talking to both ICER and IIT. And yesterday you saw when I said it's ICER means governance, administration, and research, because the bosses also have to take a call. And fortunately, in my short interaction with them, it's very clear that they are very, very open to that. And a lot will depend on the kind of enthusiasm which you people and all those who are listening to me and all those who will get communicated, that the first steps have to come from the people and from the students, from the researchers. I'm very sure the faculty will never be wanting, but together we can move. So thank you once again. Thank you for this opportunity. We can hear our chairman of the, sir, you have to respond to this issue of permission. Even 2 lines will be okay. If you give that assurance that any paper comes to you, you will. Sorry for putting that spotlight on you. No. All the young students are waiting for that assurance.
So, good afternoon. So even the DFO, Pravin Kumar, he told me that so many permissions are given. The current HOFF, that is Head of Forest Force, Pradeep Kumar, is very liberal in granting permissions. But after granting permission, they don't know. So what is happening? So that complete schedule of our activities can be given to the concerned DFO or the authority. And as Sir has already explained, so what they need is information from our side. We take technical expertise. They have a lot of knowledge that is accrued over a period of time, but they have not published papers. That is there in their minds only. So our knowledge is not in letters. So it is there in the experience also. No, the current people also, they are in the job. No, we cannot say that they are outdated. No, sir. So many times the kind of clues we get, no, sir, they are very useful, very relevant also. Only thing is that they may not express in the language that is required to publish a paper. So they'll be crude in expressing things. It's not like that. So whatever that is required by the forest department, like training, giving regular feedback, or informing in advance about our tour plan to the DFO through email or WhatsApp, that is required. So as Sir told, this personal relation is very much important. Even there's a thin line separates doing and undoing. So we can go on positive side, inform them, take them into our confidence. So once this starts, they also feel happy that their work is being projected outside. They are also being talked outside. So what is required for a human being is recognition. That is innate requirement of every being. So people feel that however small one may be, that he is recognized or she is recognized, our work is recognized, they feel happy. So this only small human dimension which we can make use of that. So as long as I am there, I can assure you that permissions, I'll help in getting permissions for the researchers.
Thank you all. Thank you again, Professor Mathur, and thank you, Sri Reddy, for being here and staying with us for this really long event. So yeah, we'll try and put up everything on the website as soon as we can after a round of editing. But maybe we can even think of a longer set of proceedings from this event to keep the momentum going. Yeah. So thank you, everyone. And lunch is waiting.